First thing I was going to do, we were putting neurons up on the board last time, and then below that we were drawing those action potentials, and I thought it might be worthwhile to try it this way. Let's kind of put one of those action potentials up, and then I'm going to graph some neurons on top of that. Never really done this before, so I don't know how this is going to work. Let's see what this looks like. We know down here there's time in milliseconds. There's plenty, and up here we're putting our membrane potential. That was the charge of the plasma membrane of that cell. We said there was a special charge around -55. We called this our threshold value. I'm going to stretch this one way out. And underneath that, we said we were getting more and more negatively charged inside the cell. So a -70 was important. That was our resting membrane potential. Anywhere above this line, we were creeping towards positive membrane potential. We'll put -30 there. We'll put 0. That's probably fine. We'll just say +30 at the top of that chart. And when we were graphing this electrical activity, I think I was using a blue color to show what we were calling resting membrane potential. When the cell was, the cytoplasm of a neuron was at rest. It wasn't sending any electrical current. When it's just at rest, its cytoplasm is got about a -70 millivolt charge inside of the cell. And what we were seeing very quickly is that there would be a slight drift towards the positive end of this graph that we're drawing here. This was, how can I draw this? This was really due to sodium ions moving into the cell. So we'll say into the cell, causes the cell to take on these positively charged ions, and the cytoplasm of that cell started drifting towards the positive end of this graph. If it wasn't a sufficient amount of sodium ions rush in that we got all the way up to -55 millivolts, then we said the sodium-potassium pump would just bring us right back down to resting membrane potential. So I'll credit this little color here with the sodium-potassium pump. So if we're too far, just slightly above or slightly below resting membrane potential, this sodium-potassium pump will bring us right back down to normal resting membrane potential. So here we are back at rest. And the second time we do this, let's take one of these slight depolarizations that this time is going to be sufficient to get all the way up to threshold. We did a couple of drawings last time because there was something special that happened as soon as we crossed threshold that now is going to involve other types of channels. And as soon as we cross threshold, the rest of the graph would immediately spring up to +30. We would see it drop down to below -70 for just a little bit. And then anything slightly above or slightly below -70 millivolts is going to be balanced out by that sodium-potassium pump. So bring us right back to this normal resting membrane potential. So nothing's happening here. I stretched this out so that we can see several of these action potentials. So I'm going to draw another one of these slight depolarizations that crosses threshold. So we have this rapid depolarization followed by what we called a repolarization. Then it drops below -70. This is our hyperpolarization. And then that sodium-potassium pump brings us right back to resting membrane potential. Let's see if we can squeeze one more in there. We've got a slight depolarization that takes us all the way up to +30, followed by one of these repolarizations, a brief hyperpolarization, and then sodium-potassium pump bringing us right back up to normal resting membrane potential. Okay, I'll just leave the rest of it like that at rest. We've got 3 action potentials that are drawn in here. I wanted to show this consecutive ways. I used different colors last time. I might have used this orange color to show that anytime we get above threshold, that this whole part of the graph where we have this steep depolarization followed by a repolarization and a hyperpolarization, this whole part of the graph is an action potential. Action potentials are always going to involve voltage gates. Oh, that's, I guess, where I'm taking away some of my room to draw those neurons at the top. I'll use another color, maybe this one real quick, just to show this electrical signal when we were below threshold. This whole part of the graph, even right over here, this is what we would consider a graded potential. So I was trying to do this last time by drawing several neurons across the top, and I'm doing it here by using the electrical activity to show that kind of what I was trying to show last time, that you've got these graded potentials followed by these action potentials, and these action potentials may lead to another graded potential followed by another one of these action potentials. So anywhere where we are involving these voltage gates, we've got an action potential. Anywhere we are involving these chemically gated channels, we've got a graded potential. I was doing that last time. Now that I've got some action potentials drawn on here, let me get that out of the way. We were drawing types of neurons last time, and if we're thinking about one of these action potentials occurring, that has to begin at this space that we called the axon hillock. And then you would have an axon that would go down and lead to an axon terminal. There would be vessels in there of neurotransmitter. Here's where we have our graded potential. So let's draw the cell body and all of the dendrites. In this area, again, cell body and these dendrites, they're covered in those chemically gated channels. I guess if my chemically gated channels represent whatever purple color this is, I'm going to put chemically gated channels all over the dendrites and all over the cell body. But as soon as we get to this part of the neuron cell body, this axon hillock, those chemically gated channels go away, and for the first time we see our voltage gated channels. So I'll put a string of voltage gated sodium channels. We'll put our string of voltage gated potassium channels. And then finally, those voltage gated calcium channels are on the ends of that axon terminal. We've set up our chemical gates versus our voltage gates.
What were the little purple things on the dendrites called?
I'm trying to use this color to represent my chemically gated channels. So chemically gated channels are going to be located on the dendrites and on the cell body of neurons. And when we graph them, we're graphing them in this part of the graph that are below threshold, because we're not involving any voltage gated channels. Right? Without using too many colors, if I can just put a dotted line here. On one side of this line, we've got the dendrites and the cell body and all of the chemically gated channels. And on the other side of this dotted line, we've got what we call the axon. We've got the axon terminal. And both axon and axon terminal are covered in these voltage gated channels. So I've used 3 different colors to show voltage gated channels for sodium, voltage gated channels for calcium, or those voltage-gated channels for potassium. Sodium ions, potassium ions, calcium ions, they have these voltage gates, and those voltage gates are located physically in a different part of the neuron. From the axon hillock to the axon terminal. And anytime we're involved in those voltage gates, that was this part of the graph. We put a couple of these little asterisks last time. The other one was right here at -70. Because these were the triggers. If we crossed these little triggers at -55 millivolts and then on the way back down at -70 millivolts, these were the triggers that changed the positions of those voltage gates. So the end of our lecture last time, we were talking about why it is that you have this absolute refractory period. That once you start one of these sodium voltage gates opening, it's not going to be able to send another action potential until it resets. So after it resets and drifts below -70 millivolts, you've got this brief, what we called a relative refractory period, because the voltage gates have reset, but we're further away from threshold than we normally would be at resting membrane potential. So after everything resets, now we can send another action potential if a sufficient threshold stimulus is reached. We said an insufficient threshold stimulus is never going to generate one of these action potentials, because that would be, again, here we are, and that sub-threshold stimulus, our analogy is we're standing on a dock, right, throwing little pebbles into the water, and in this case it's a neurotransmitter, a chemical signal that's landing on these chemically gated channels. If it's just a little bit of neurotransmitter, we'll zoom into this because we're about to start with a video that shows these chemically gated channels. We've seen them before. We called them non-specific ion channels. Non-specific ion channels, they're going to let sodium ions move into the cell. And it's true that some potassium is going to leak out. Again, it's not specific which ions move through, but more sodium is going to move in than potassium is going to move out. So you've got this local wave of depolarization that's kind of spreading across this cell body. And if we're just throwing these tiny little pebbles, then maybe this wave of depolarization never hits those voltage gates, never gets all the way up to threshold stimulus. But again, if we're throwing enough of these neurotransmitter rocks, in my analogy, that there's sufficient amounts of sodium ion that comes in, this wave of depolarization is going to tip that first domino over at the axon hillock. Now we are at this part of the graph where we're involving those voltage gates. And I won't do the same drawing again unless we have review questions about it, where there were 3 positions of that voltage-gated sodium channel. It opened very quickly and went through a series of open and 2 positions in which it was closed. Again, it had to be reset if we were going to generate another one of those action potentials. So we tried to make a distinction last time. I'll use a red color here. Between right here what we called our absolute refractory period versus this part of the graph where our voltage gates have reset. They have gone and repolarized past -70 millivolts. So they're capable of sending another action potential, but just for a brief period of time, we're going to be even further away from threshold than we would be at normal resting membrane potential. This is our relative refractory period. Okay. I wanted to go through this quick review again just to point out the anatomy of these neurons, the location of chemically gated channels on the cell body and the dendrites, and then the location of these voltage-gated channels on the axon and the axon terminal. In fact, you can see on the graph here, we're about to get into another one of these graded potentials. So that means we would have one of these neural synapses. We can draw another one of these neuron cell bodies with a long axon. We'll make that an axon terminal. And if we're thinking of a graded potential, that's going to involve, once again, these chemically gated channels. And some type of chemical signal docking to these chemically gated channels. This chemical signal is going to be neurotransmitters released from the axon terminal. There they are. So just for reference, I wanted to have two neurons talking. We'll spend a little bit of time going through this again. And our terms in referring to neural communication, we've got our presynaptic neurons sending this signal to a postsynaptic neuron. We said neurons were like one-way streets. So it's always the dendrite or the cell body that receives a signal from another neuron, and that signal is going to leave out of the axon end. You never have an axon end receiving a signal and sending it back towards the cell body. Again, it's like a one-way street. Dendrites and cell bodies receive signals. Axons send them away. Any questions or thoughts about that diagram?
Yes.
Yes, what's up?
Depolarization that's created
Yeah.
the sodium entering into the cell.
Yeah, let's label that.
Repolarization is the purple, and what is that?
That's right. So when we're graphing all of this stuff, these waves that go up and down. So this part of the wave, as soon as we are, anything that takes us from our normal resting state, negative 70 millivolts, and drifts us up closer towards this zero membrane potential, anything in green is what we call our depolarization. And we said this depolarization is due to the fact that you've got sodium ions moving into the cell. So the cell's taking on all of these positive charges. And then the other side of that graph we said was repolarization. Repolarization, as we're seeing here, the cell is becoming more and more negatively charged inside, and that's due to the fact that it's getting rid of those potassium ions. So potassium ions are moving out of the cell. We graphed this last time. We said neurons, they have inside of them a bunch of these negatively charged proteins. And if you look at this model here, it's got all of these blue proteins that you see just kind of floating around in the cytoplasm. Those are those big negatively charged proteins. Where are we? So if the cell can kind of purge itself of those potassium ions, it's leaving behind just a bunch of those negatively charged proteins. And there was something about that potassium gate that we said was slow to open. It was told to open right here, but it actually didn't open for a few milliseconds because it was like those apartment gates. And it's also slow to close. So it's told to close right there, but again, there's a few milliseconds until it actually closes. And that excess loss of potassium ions is what we called hyperpolarization. So hyperpolarization, once again, due to the fact that you got potassium ions moving out of the cell. And let's not forget the work of that sodium-potassium pump, always helping reestablish that resting membrane potential. That's right. So any part of this graph where we're involving these voltage-gated channels, this big sway in membrane charge, that's a rapid charge of about 100 millivolts, we said, was our action potential versus these graded potentials that occur on dendrites and cell bodies. Trying to connect the dots with this electrical activity that we were drawing and where it's occurring on these neurons. And I was inviting any questions that you might have had or thought about since the last time that we met. Okay, let's see. Most of this, like I said, we've already talked about. We should be able to cover a neural synapse. So this neural synapse is the thing that we were just sketching on the board. We've got a little bit of it left between the axon terminal of one neuron, and then we can see the dendritic end of a postsynaptic neuron. And both of the class handouts, I'm going to grab this one again, the neural tissue one that we went through last time, the very back of that, you've got this same picture. You've got a presynaptic neuron talking to a postsynaptic neuron. And even on the second class handout, this neurophysiology class handout, you've got the detailed picture of this chemical synapse. You've got a presynaptic neuron talking to a postsynaptic neuron. We should be able to once again detail that today. That's really the main point of today's lecture, to make sure that we've covered that physiology discussion last time. How is it that these neurons are capable of sending these electrical signals? Where do they get that electrical current from in the first place? We've compared graded potentials versus action potentials. So far, we've really only talked about one neurotransmitter. I'm looking for this color because I've been drawing these little blue dots on the board that represent acetylcholine. I'll just circle this as a common neurotransmitter that's used by neurons. By the end of this semester, we'll see that neurons can use different types of neurotransmitters, and they're going to cause different types of signals. They can either be excitatory signals or inhibitory signals. Let me see if I can cross this off the list right now. EPSP versus IPSP. What I've drawn right here, this little green arrow or line that starts to cause the cell to become more positively charged and drift closer towards threshold. This is, get my colors over here. This little bit of the graph that caused the cell to become slightly more positively charged, this would qualify as an EPSP. This just stands for excitatory postsynaptic potential. Remember, in our neuron physiology discussion, we've got a presynaptic neuron and our postsynaptic neuron. And our question here is, what's it going to take for this postsynaptic neuron to send an action potential? And I've kind of led to that earlier. You know, imagine that there's another person standing on one of these dendrites, and those chemical signals are the little pebbles that we're throwing onto the surface of the water, and we're trying to create a large enough disturbance that this wave of sodium ions is going to make its way all the way over to that axon hillock. I know the screen is blocking this second neuron, but what we've got to do is we've got to open up enough of these chemically gated channels that sodium ions rush in, and that wave is going to tip over that first domino at the axon hillock. Excitatory postsynaptic potentials take you closer towards threshold. If we were to go the opposite direction, I don't have enough of it left over there. But if this signal were to, instead of taking us closer to threshold, take us down to negative 80 millivolts, somewhere further away from threshold, this would be what we call an IPSP, or inhibitory postsynaptic potential. So this postsynaptic neuron can either be hyperpolarized and pushed further away from threshold, or it can be stimulated and driven closer to threshold. Here's an example. You're grabbing on any of the reflex arcs. I think I mentioned slamming my hand in a car door or stepping on a tack. If my hand is getting slammed in the car door and I need to pull my hand away, there is a signal, an excitatory signal that's sent to the biceps to get me to pull my hand back. But if I'm going to flex my arm, if the biceps is going to fire and contraction is going to occur, and my arm is going to flex, I have to have at the same time a signal sent to the triceps that's inhibitory, that prevents it from contracting, causes it to relax. So neurons simultaneously will be sending excitatory signals in one direction and inhibitory signals in the other. Again, we'll compare IPSPs that are capable of drifting up to threshold and generating an action potential, and then these IPSPs that basically prevent an action potential.
So
correct.
If it goes up, that's no longer it.
That's right. So we're going to be dealing with these chemical signals on dendrites that are either going to open or close gates. You can imagine some of these neurotransmitters, if they allow sodium ions to rush in, it's going to be an EPSP, because as sodium ions rush in, we drift closer to threshold. But other neurotransmitters are going to cause potassium ions to leave. Just a few potassium ions to leave rather than sodium to come in. So now this cell body is going to hyperpolarize. It's going to get further away from threshold. So it's interesting that just by controlling these gates, you're going to be able to control whether or not neurons can send signals or not. Hopefully we're doing all right.
I'm sorry, one more.
Yeah, no, go for it.
Hyperpolarization, that wouldn't be an IPSP?
So an IPSP is going to cause a cell to hyperpolarize.
So like where it says relative refractory period, would that be IPSP technically?
Well, this wouldn't be an IPSP because this hyperpolarization is due to the fact that the voltage-gated channels have been triggered and they're slow to close. So this is following the back end of an action potential. So that hyperpolarization would be due to slowly closing voltage gates. This hyperpolarization is going to be due to chemically gated channels on the dendrites of the cell body that are going to prevent this voltage-gated channel from ever getting triggered.
Thanks.
Yeah, no problem. Hello? We just put a review of the diagram that we had up on the board last time. I was inviting questions. As I continue to invite questions, we're just going to click through a few of these slides from the neurophysiology PowerPoint. And this is actually the first slide, so you're kind of coming in just at a good time. And I can click past this slide because I've already drawn this on the board. We should be able to recognize this structurally as a multipolar neuron. It's got multiple dendrites attached to this cell body. And like we just drew on the board, we would know that there are chemically gated channels all over the dendrites in the cell body. Here's where we have our voltage-gated channels down the axon, all the way to the axon terminals. And we talked about how these action potentials are generated when those voltage-gated channels are triggered. Here's what we've, again, drawn a few of these on the board. This slight depolarization, another way of describing what just occurred right there is an EPSP. Right? There's an excitatory signal that has caused our cell to take on a few sodium ions. It has drifted closer to threshold. It's a slight depolarization. I know I put some of these same slides in that Google slide deck that I sent out. So you've seen what this slide is trying to describe. It's trying to describe how these neurons have a negative charge to begin with. How are they at negative 70 millivolts normally when they're at rest? And we looked into this where if you look inside the cytoplasm of a neuron, we said it has a bunch of these negatively charged proteins. In addition to having negatively charged proteins inside the cell, it has a bunch of potassium ions and very little sodium ions. Versus outside the cell where we see potassium levels are very low and sodium levels instead are very high. This imbalance between sodium and potassium, it is established by the sodium-potassium pumps. So we spent some time graphing the different positions that sodium-potassium pump can take, either throwing sodium ions out of the cell or pulling potassium ions inside the cell. We said once that sodium-potassium pump works for just a little bit of time, it changes the concentration of sodium ions to being extremely high outside the cell and very little sodium ions inside the cell. Versus those potassium ions, we see very little of them outside the cell, but a lot of potassium ions inside the cell. We jump back there for a second. The other thing to note, those big negatively charged proteins, they're too big to leave the cell. So you don't find any of them in that extracellular fluid, but there are plenty of those negatively charged proteins inside the cell. So this imbalance of negatively charged proteins is one of the things that contributes to our resting membrane potential, this negative 70 millivolts. There was the video last time that showed these two positions of the sodium-potassium pump. We can see when it's open to the inside, sodium is able to dock. Once sodium loads all the way up, it gets phosphorylated. A little phosphate gets stuck to it, causing it to change positions. Now it's open to the outside. And we said when it's open to the outside, those docking sites for sodium immediately change. So sodium can no longer stay there. It's kicked off. It's ejected. And at the same time that these binding sites for sodium change to no longer let sodium bind, those binding sites for potassium have now opened up. Back there, those binding sites for potassium were closed off. Potassium couldn't bind, but simultaneously they open as the ones for sodium closes. And now potassium is jumping over here. Now potassium is able to load up. Once it fills both of those binding sites, the phosphate has been removed, causing that sodium-potassium pump to change back to its original open to the inside position. When it's open to the inside, those binding sites for potassium close off and we see potassium leave. We discussed this a little bit last time because the series of pictures is kind of a slow-moving series of events, but there was the video if you watched the Google Docs or that slide thing that I sent out. I'll be glad to give more examples of that if we need. We looked at, we listed in fact over here different types of channels last time. We said there were leak channels on neurons that are always open. I'm going to use this color to just draw a typical neuron. So our neuron plasma membrane, to the outside we've got our extracellular fluid. To the inside we've got our intracellular fluid, or our cytoplasm. And what we know, we were kind of looking a little bit about this last time. There's going to be a bunch of different proteins embedded in the plasma membrane. And I'm going to start here at the beginning by just putting in these leak channels. Leak channels are just like straws. They're hollow tubes. They're always open. Let's list this as the leak channel. And if leak channels were the only channels that were around, what you would have, potassium ions. If we had a bunch of potassium ions inside the cell, if we had more of them inside the cell than we had outside the cell, these channels are constantly open, so potassium would just drift to the outside until there was equal numbers of potassium inside and outside the cell. Let's see, 1, 2, 3, 4, 5, 6, 7. So we got 3 more to go. I think we're good. Now that we've got equal numbers of potassium inside and outside the cell, there is no net movement of potassium. There may be one drift in or maybe one drift out, but they're staying at equal concentrations. These leak channels are not specific to, I'll say it this way. There are leak channels that are specific just to sodium and leak channels that are specific just to potassium. We don't have to get to know them in that great a detail. The point that I'm making about leak channels, whether it's sodium or potassium leak channels, is that they're always open. And if they're the only channels that existed, we would see that there would be an equal number of sodium ions both inside and outside the cell. We know there's a lot of sodium ions outside the cell. How many do we got there? 7. If leak channels were, again, the only channel open, we would see that they would allow the equal distribution of sodium ions to, if we had more sodium outside the cell than we had inside the cell, then we would see sodium start to drift inside. So thanks to the movement of these leak channels kind of always being open, it allows sodium and potassium to kind of reach this equilibrium where there's the same number of sodium outside versus inside the cell. We could say the same thing about potassium. Leak channels. Again, the thing that battles this even distribution of sodium and potassium, we put them up last time as little red circles. This can represent our sodium-potassium pump. We said the sodium-potassium pump works to establish this concentration gradient where it's grabbing any of these sodium ions inside the cell. It's taken 3 of these sodium ions from inside the cell. So 1, 2, 3 sodium ions, and has thrown them out. At the same time, it's going to take 2 of these potassium ions from the extracellular fluid and bring them back in. I'll take that one. This sodium-potassium pump is doing the same thing. It's throwing 3 sodium ions outside and 2 potassium ions back into the cell. So let's take 2 more potassiums out. We'll take 3 more sodiums out. Let's put those other sodiums out here somewhere. So we can see we've got a really high concentration of sodium outside the cell, and all of those potassiums that we were grabbing from outside ended up in here. There should be 4 of them that we get to add. So with that little quick review, you really get to see how the working of these sodium-potassium pumps creates this concentration gradient of high concentration Just put this out here. High sodium outside. Versus low sodium in the cell. We contrast that with very high concentrations of potassium ions inside the cell and very low concentrations of potassium outside. Thanks to that sodium-potassium pump. So now we've basically set the stage. What we haven't really talked about yet and where we're going in the next couple of slides, and I've already kind of used these same colors, but the other channel that we're going to talk about are these voltage-gated channels for sodium. And then these voltage-gated channels for potassium. So they're not an open tube. You can see they're closed off. They've got these gates that when the cell is at rest, these gates are closed. But kind of going back and forth between those diagrams and what we are drawing here, if any of these voltage-gated sodium channels were to automatically open up, which happens when these cells depolarize past threshold, you can see it's going to be very easy for a lot of sodium ions to rush into the cell. I drew one through that leak channel, but I meant to draw them through these voltage-gated sodium channels. We said sodium rushes in really for two reasons. There's a concentration gradient difference of high sodium outside and very little sodium inside. And the other thing that really pulls sodium in is the fact that we've got all of these negatively charged proteins inside the cell. They're not outside the cell. Those positively charged sodium ions like these negatively charged proteins. And that's one of the things that helps pull sodium into these cells very quickly. That's what we called our depolarization. I'll just come back over here and represent it as a depolarization. Now we've got a bunch of sodium that is rushing into the cell, and that's fine. We still have very little sodium outside the... Let me say that again. I just had a mutation of words. A bunch of sodium coming out. All right, what I was just showing here is that sodium rushes into the cell very quickly because there's lots of sodium outside the cell and very little sodium inside the cell. But now let's switch and think about potassium, the purple one. There's lots of potassium inside the cell and very little potassium outside of the cell. If this cell needs to repolarize, it's going to be easier for it to get rid of potassium. We've got this concentration gradient built up of lots of potassium inside, very little potassium outside. So as we hit the peak of this action potential and those voltage-gated sodium, the voltage-gated potassium channels finally open up to the point where potassium ions are now, so once these voltage-gated potassium gates finally open, now potassium, it's all spring-loaded. It's ready to fly out of these cells. Go through the right gate. This cell getting rid of all of these potassium ions is our repolarization. I mentioned that in repolarization that you've got really potassium ions leaving just because of the concentration gradient difference. They're going against the electrical gradient here. Those positively charged potassiums, they like the negatively charged proteins, just like the positively charged sodiums also like those negatively charged proteins. Potassium is going against its electrical gradient, but down its concentration gradient. Sure.
How is the low sodium and how is it low sodium and high potassium in the cell?
So that's why I was doing this kind of in a series of stages. And do you remember before I put any of the sodium potassium pumps up there, and it was only those leak channels that were like hollow straws, then there was an even distribution of sodium and potassium on either side of that membrane, right? But then what we did is we put on those sodium potassium pumps, and they started to work by grabbing sodium ions that were inside the cell and throwing them outside the cell. And if we let that, this is kind of where I was over here. Remember, the sodium potassium pump will work to reestablish this negative 70 millivolts. If there happens to be too many potassium ions into the cell, or if there's I did it again. If there happens to be too many sodium ions into the cell, or too many potassium ions leaving the cell, if there's a slight imbalance of sodium or potassium, this sodium-potassium pump can reestablish that concentration gradient of lots of sodium outside and very little sodium inside the cell. Same thing with potassium. That sodium-potassium pump pulls in a lot of potassium as it's getting rid of that sodium. So potassium doesn't come back in to make it more negative, but we would see potassium is purged to make it more negative. Potassium's got that positive charge too. So that's what was kind of counterintuitive to me when I was first learning about these action potentials, that for a cell to become more positively charged, it takes on a bunch of sodium, but then to become more negatively charged, it gets rid of a bunch of potassium. Right? You would think it just took on all this sodium. Why is it not just getting rid of the sodium? But there's already a bunch of sodium outside the cell. It would be harder for sodium to leave the cell than it would be for potassium to leave the cell, because the sodium-potassium pump has already removed a bunch of potassium from that extracellular fluid. So that potassium is set to spring out of the cell faster than sodium would spring out of the cell. So it depolarizes with sodium coming in, and it repolarizes with potassium going out. I can finish labeling my graph over here. Now that we've got our depolarization with sodium going in, we've now got that followed by repolarization. This part of the graph where potassium ions are moving out. Same thing down there with our hyperpolarization. We spent a little bit more time last time talking about this hyperpolarization because the fact that those voltage-gated sodium channels are very quick to open and quick to close, but the potassium channels are slow to open and slow to close. Let's catch back up with our slides. And again, throw out any questions if you're thinking about them. Leak channels, yeah.
Do we have depolarization or repolarization?
Depolarization, that's right. So depolarization. So below this threshold, we've got a slight depolarization that's due to these chemically gated channels, and that's this depolarization that I'm drawing in, I guess, the blue color here. You know, depolarization is sodium ions rushing into the cell, causing that cell to become more positively charged. In the graded potential world, where we are down here, you know, this depends on how much neurotransmitter is present. So it may or may not ever reach over to that axon hillock. If it does, then what we've done is we've graphed this part right here where there's enough of these sodium ions, this depolarization due to chemically gated channels, that we've crossed over threshold, and at our little trigger point here is when we start to involve those voltage gated channels, that first domino that falls. And now we're graphing our action potential. So you're right, sodium ions rushing into the cell. This is all a depolarization. And anytime the cell is becoming more negatively charged over here, the cytoplasm of the cell is becoming more negatively charged, then that's our repolarization, and that's potassium going out.
So why is it easier for potassium to leave the cell in repolarization than it is for sodium to leave the cell?
So right here, and this is where, you know, if we could expand this diagram out and I could draw more sodiums all over the place here, you would see that we did have an influx of sodium right here along the surface of the membrane, but that's backed up by stacks and stacks of sodium. So it's still going to be, you know, if those voltage-gated channels were to stay open, sodium would keep moving into the cell. So to prevent sodium from moving into the cell, the thing that happens very quickly, as soon as we reach that plus 30 millivolts, those sodium gates close. So no more sodium rushes into the cell. There's just such a high abundance of sodium in this extracellular fluid that just a little bit rushes in, and then at the peak of that action potential, those voltage gates stop that sodium from rushing in. So we have more sodium in here than we normally would, and that has caused this depolarization. But still, if we were to open up those voltage-gated sodium channels, even more sodium would rush in. So if we want to instead get rid of positive charges, the only option we really have is to instead deal with potassium ions that we've been adjusting the concentrations of as well thanks to that sodium-potassium pump. It's been working to remove all of those potassium ions from the extracellular fluid and really build them up into the cell. So it's interesting that the neuron membrane doesn't care whether it's sodium ions or potassium ions. It's just it takes on a bunch of positive charges for depolarization, and then it gets rid of a bunch of positive charges for repolarization.
Okay, so it's like they open at the same time, but sodium gates close sooner at that like peak point in the.
That makes me want to draw that one diagram again, where it's the same trigger that tells both of those voltage gates to open, but it was only the sodium gate that opened very quickly.
The potassium takes longer.
Yeah, it's like the apartment gates. It was told to open at the same time the sodium voltage gate was told to open, but that sodium voltage gate kind of opened immediately, and there's just a few milliseconds where it's only sodium that's able to cross. So sodium rushes in very quickly until we hit the peak of that action potential, and that positive 30 charge is what closed off those voltage gates. So sodium just rushes in very quickly for a brief period of time, and now that allowed enough time for those voltage-gated potassium gates to open, and now potassium floods out of the cell. So if you're thinking of it, I'm sure 5 other people at least are thinking of it too. So definitely let me know if there's questions or anything you're thinking about with it. I was going to try and catch up with the slides because we know it's not all about these leak channels. If it were only leak channels, there would be even concentrations of sodium and potassium on either side of the cell. But we've got chemically gated channels, which we know are located on the dendrites in the cell body. We've got voltage gated channels that are located on the axon and the axon terminal. There is even another one. I'll sketch this for you real quick and we'll see it later. There's going to be something called a hair cell. We'll see these when we talk about special sensory. And a hair cell looks something like this, where it's got this plasma membrane extension that extends out something like this. This is the nucleus of the cell. Here's our hair cell. It's got these hairs that stick up. And on one side of the cell, so imagine this has a bunch of potassium ions inside the cell, a bunch of these sodium ions outside this. Draw that sodium. A bunch of these sodium ions outside the cell. In mechanically gated channels, the gates themselves are going to be located on one side of these hairs. Just put a few of them in there. And maybe I could have zoomed in a little bit on this. Remember those little tiny coiled proteins that we were drawing as the titin protein? Those titin proteins come back and they basically anchor to one side. They're attached to this channel and then they spring over to the other side of this hair. So it looks something like this. Again, our coiled titin protein is nestled on one side of the hair. It's attached to this bristle. On the other side, it's attached to that mechanically gated channel. And the whole thing is set up so that if we were to bend these hairs over in this direction, it would pull tight on that stringy titin protein, and it would do just like I'm doing with this door. It would physically open the door. Or you can imagine if we were to instead, this would trigger an EPSP. If you bend it in this direction, it gets all excited. Those gates are pulled open and sodium rushes into the cell, versus if you were to bend those hairs over the opposite direction, this would inhibit those mechanically gated channels from opening at all. It would be like me holding this door shut and preventing any oh, sorry, see? I was just showing these mechanically gated channels are either forced open or held closed. All right, we have some close-up pictures here of the different types of channels. I feel like we've talked about these enough here. Chemically gated channels like we're seeing here, they either open or closed based on the presence of some type of neurotransmitter. So if no neurotransmitter is docking to them, they're in the closed position. If we do have neurotransmitter, then they're open, and you can look, here's that non-specific ion channel. It lets both sodium and potassium cross. We know more sodium comes in than potassium leaves, so these lead to a slight depolarization. Then if we get to the axon hillock portion, we've got voltage-gated channels that open and close based on the membrane charge. Okay, so we've graphed this. Depolarization is when we're drifting closer to threshold. I left some of these slides that have just words in them so that you can click through them if you're just doing this at home. But I've said all of these things. When we're thinking about depolarization, we've got a reduction in the plasma membrane's charge. It's creeping closer towards zero membrane potential, closer towards the positive end of this range. Again, we've seen that picture plenty of times. And also look, since we are below threshold, look at where we're at. We're over here on the dendrite or the cell body end. We've not yet involved any of those voltage-gated channels. Hyperpolarization down here, this is when there has been an excess loss of potassium ions. Again, we're further away from threshold. We've lost excess potassium ions. All right, again, there's our first picture of our hyperpolarization. That would be due to an inhibitory signal.
So after the hyperpolarization, the way that gets fixed back to resting membrane potential is the sodium potassium pumps bring in.
That's right. That sodium potassium pump would say, oh, we're slightly off from there. We've got too many potassium ions that have left the cell, so it would pull some of those potassium ions in and get rid of sodium. Helps maintain that resting membrane potential if we get too far away. So there's our first inhibitory picture. I feel like we've seen this with neuron function already. So where are we going? Action potentials versus graded potentials. We've spent some time setting this up, and this dotted line that I drew on the board is what I was attempting to show on this next graph. Back when we were just learning from home and I couldn't draw anything on the board, I had to just put this little blue line across this picture of a neuron. But you can see it's trying to diagram what I've been saying since the beginning of this lecture. If we think about the cell body or the dendrites, this is the area where you're going to find chemically gated channels. This is the realm of graded potentials, because the strength of that electrical signal depends on how much chemical signal is being used. Versus the action potentials that are all of these voltage gated channels that open and close based on specific voltage signals. So action potentials are always the same strength. They're the same triggers that cause those gates to open and cause those gates to reset. So there's no varying in strength when we're thinking about action potentials. But graded potentials, there is varying in strength depending on, again, the varying amounts of neurotransmitters that might be there. Okay, so different parts of the neuron. Graded potentials, like we were just saying, these are short-lived chemical signals. They may cause, like we were showing right here, a slight depolarization or a slight hyperpolarization. But if we don't cross threshold, we're never involving these voltage-gated channels. It's just still the realm of chemically gated channels. Scanning for questions. Let's see. At this point, I'm going to play this video again just to see if maybe this will jog some questions. We saw this last time, but it's where we're going. It's the details of that neural synapse between a presynaptic neuron and a postsynaptic neuron.
A chemical synapse is a special junction between two neurons that allows them to communicate without being physically connected. A synapse has several structures. The presynaptic neuron ends in a small bulb called the presynaptic terminal. The postsynaptic terminal membrane is directly adjacent, separated by a small space called the synaptic cleft. An action potential causes voltage-gated calcium ion channels to open in the presynaptic terminal. The influx of calcium ions prompts synaptic vesicles to release neurotransmitters via exocytosis. This neuron is releasing acetylcholine. The neurotransmitter quickly diffuses across the synaptic cleft. Two acetylcholine molecules bind with one ligand-gated sodium channel at the postsynaptic membrane, opening the channel. When enough sodium ion channels open, the postsynaptic cell depolarizes and the action potential continues along the neuron.
Remember, ligand-gated channels are the same thing as chemically gated channels. And like we were seeing just right there, if there was enough chemical signal present that those non-specific ion channels were open for long enough, then that allowed the wave of sodium ions to rush in to eventually get all the way over to that axon hillock, and we can see send a signal down the rest of that neuron. Let's see. So here's the picture that you have on your lecture handout, that second one. And it's the video that we were just looking at kind of detailed each of these steps. There was a signal, an action potential coming down the axon of the presynaptic neuron. And it's really this electrical signal that first opens up these voltage-gated calcium channels. We've seen this now multiple times, all the way back to that neuromuscular junction. The action potential, once it hits that axon terminal, we said it opens those voltage-gated calcium channels. Calcium then moves into the axon terminal and surrounds those synaptic vessels. Normally, those synaptic vessels, their membranes are lined with a slight negative charge. Just like the membrane of the plasma membrane here has a slight negative charge. So those negative charges typically oppose one another, and these synaptic vessels just kind of bounce around in the middle of this axon terminal. But when calcium ions come in and they surround those vessels, they're now coated in a positive charge, which is drawn towards that plasma membrane. So we said when calcium ions move in, that leads to the release of neurotransmitter. So neurotransmitter is released into the synaptic cleft. We can see it takes a little bit of time to diffuse across this synaptic cleft and land on that receptor. That time that it takes for the neurotransmitter to diffuse across that little synapse is what we call synaptic delay. Anyway, I'll leave that there. Going back to this, that synaptic delay again was the few milliseconds that it took for the neurotransmitter to diffuse across the synaptic cleft, land on those receptors. We can see this is our postsynaptic neuron. We've got chemically gated channels located on the cell body and on these dendritic ends. And these are non-specific ion channels, so that when a neurotransmitter lands on it, you can see, being a chemically gated channel, it opens. It allows sodium to come in and potassium to go out. It stays open as long as the chemical signal is present. If for some reason that chemical signal is removed because it diffuses away, or maybe there's one of those enzymes like acetylcholinesterase that breaks down that chemical signal. Once that chemical signal is removed, we can see the chemically gated channel closes. So depending on how much neurotransmitter is present, if there's a continuous shower of neurotransmitter, then this chemically gated channel is going to open back up. There's going to be even more sodium that rushes into the cell. It may or may not be sufficient to get all the way up to threshold. We saw synaptic delay. This is just a closer picture of one of those non-specific ion channels. Again, when the chemical gate is open, this is allowing sodium to move into the cell and some potassium to move out. Once removed, this is done for a few reasons. There is, like I mentioned, an enzyme that can remove this neurotransmitter, close that channel back. There is a possibility of this neurotransmitter just diffusing away from the receptor. It may not be broken down by the enzyme. And if it does diffuse away, one possible I don't know what I'm losing a descriptive term there. One fate, I guess, of these neurotransmitters that diffuse away is they can be actually taken back in by the axon terminals. This reuptake of neurotransmitter is something that allows neurotransmitters to be recycled over and over. Anyway. Maybe we'll see some case studies about this, because there are some medications that will prevent the reuptake of certain neurotransmitters and cause them to have long-lasting effects in that neural synapse. Okay, so again, I think we crossed this one off the board earlier. EPSPs just stand for some type of excitatory signal that's being sent to a postsynaptic neuron. So if the plasma membrane of our postsynaptic neuron depolarizes, that is one way of describing one of these EPSPs. In fact, I think there are some questions, and we can practice doing this right now because we were doing this just earlier when we were going through some questions. We said an EPSP, we can describe an EPSP two ways. It's an excitatory postsynaptic potential. It's due to a cell depolarizing. It causes a cell to depolarize rather than hyperpolarize. An IPSP, an inhibitory signal, is going to cause a cell to hyperpolarize. And instead of using the term depolarize or hyperpolarize, we could have said it this way. We could say EPSPs, again, these excitatory signals, will cause a cell to take on sodium ions. Sodium ions move into the cell in these EPSPs. They take on positive charges. Another way of saying that is depolarize. Again, IPSPs cause a cell to hyperpolarize, but another way that we could describe that is IPSPs are going to cause a cell to lose excess potassium ions. Hyperpolarization, we've just lost excess potassium ions. And what happens is we're further away from threshold at that point. So it's going to be even harder to generate an action potential. It's going to be harder to get that first voltage-gated domino to fall if we're hyperpolarized. So this is a way of inhibiting neurons from sending action potentials, while depolarizing neurons is a way of promoting them to send an action potential. We still doing okay? Here's an example of one of these EPSPs. Right, EPSP, we can see a cell is taking on positive charges. We can describe this, like I said, two ways. This is what we are looking at here is a slight depolarization. And we know it's depolarizing because sodium ions are moving into the cell. That's what happens in our EPSPs. Versus, I'll say that again, EPSP, like we were just seeing, sodium ions are going to influx into the cell. This is what causes our depolarization. If it is sufficient, it may cross threshold and generate one of these action potentials. We said EPSPs promote action potentials if they cross threshold. IPSP is going to inhibit an action potential because it's going to push a cell even further away from threshold. So these IPSPs are inhibitory signals. They cause a cell to hyperpolarize. Again, what we're seeing here is a cell has hyperpolarized because it has purged itself of even more potassium ions. I feel like we're being really redundant there, but hopefully if we hear it enough, it will kind of start to settle in there. There are two types of summation that we need to be able to distinguish. It's not a huge part, but since we're talking about neurons talking to one another, there's two types of summation that we can point to. One type of summation looks like this. It's called temporal summation. I describe this temporal summation. There's going to be one presynaptic neuron that's going to send multiple signals to a postsynaptic neuron. So one presynaptic neuron, multiple signals. But multiple action potentials to postsynaptic neuron. Let's try and draw what that is. Let's say the blue neuron here is my presynaptic neuron. We've got one presynaptic neuron. There's its dendrites, and it's got this axon portion. And it's sending a signal, let's say at time 0. This thing has sent one action potential that measures plus 5 millivolts. And it's sending that signal to, let's draw this postsynaptic neuron in red. So here's our postsynaptic neuron with its dendrites. So we've got a presynaptic neuron talking to a postsynaptic neuron. In the case of just one signal being sent by this presynaptic neuron down to the postsynaptic neuron, this initial stimulus of plus 5 millivolts, if we remember that each one of these neurons starts at negative 70 millivolts. There's our postsynaptic neuron hanging out at negative 70 millivolts. If it is hit with this we could also describe as an EPSP. It's hit with an excitatory signal of plus 5 millivolts. That's going to get this thing to go from negative 70 to negative 65 millivolts, which we know, if we go back to our graph, is still not above threshold. So this is not a sufficient threshold stimulus. There is no action potential being sent by this postsynaptic neuron. But in temporal summation, this is what we consider multiple signals stacked together in a very short time frame. To try and draw this temporal summation, we've got multiple EPSPs. So let me add with this color, if we had, I will give myself more space. We'll say time 0, time 1, and time 2. If we have back-to-back 1, 2, 3 plus 5 millivolt signals, if they're back-to-back like that, bam, 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 5 plus 5 millivolts 3 times in a row is going to get this cell to go from negative 70 all the way up to threshold of negative 55 millivolts. And now if we get this cell to negative 55 millivolts, we're sending this action potential down the postsynaptic neuron. So temporal summation is when there are multiple signals sent from a presynaptic neuron to a postsynaptic neuron in a very short period of time, compared to another type of summation. This is spatial summation. I'll just put spatial summation down here. In spatial summation, we can define it as there are multiple presynaptic neurons. So multiple presynaptic neurons, and they each send one signal to postsynaptic neuron. So we'll put that, once again, that postsynaptic neuron, let's draw that postsynaptic neuron in red. Let's put its dendrites out there. Its nucleus is here. It's hanging out at negative 70 millivolts, just like a typical neuron would do. There's our postsynaptic neuron and our presynaptic neuron. Down here, we're going to see there are multiple presynaptic neurons. So I'm going to draw one presynaptic neuron in this color, and it's going to be sending one signal, one action potential to this postsynaptic neuron. We'll say presynaptic neuron A. It's sending its signal of plus 5 millivolts, which alone is not enough to get this postsynaptic neuron to depolarize. But if we had multiple presynaptic neurons, each sending its own signal of plus 5 millivolts, I'll just put this other one here in a black color. You've got 3 different presynaptic neurons all sending the same signal to a postsynaptic neuron. And together, this, what we would call spatial summation, is sufficient to get this postsynaptic neuron to depolarize across threshold. Okay. Here's the last new things from this slide. If we think about how neurons can code for different strength of stimulus, we're going to see that action potentials, like I was just saying, they're always the same strength. It's the same triggers that cause these gates to open versus reset and close. So strength of action potentials are always the same. How neurons can distinguish between strong signals or weak signals has to do with the number of action potentials that are generated. We can see this graph shows a sub-threshold stimulus is not going to generate any action potentials. Think of me standing over here tapping on my knee very slightly, and if I'm just very lightly tapping, that's a sub-threshold stimulus and it's not generating any action potentials. A sufficient threshold is going to definitely stimulate action potentials, but a stronger stimulus is going to generate even more action potentials. You can see each action potential is the same strength, so we can't distinguish an individual action potential from another action potential. They're all identical, but how we can make a signal stronger versus another signal is by packing in more action potentials. So you can see a stronger stimulus just packs in more action potentials than a weak stimulus. Subthreshold stimulus doesn't have any action potentials. And the last graph is something that I've already talked about, which is at least half of this I've talked about when we were thinking about the speed at which neurons can send these action potentials. How do these waves of electrical current move along a neuron? We're going to see that there are two things that affect how fast this action potential can travel. I was doing this last time by acting as the electrical impulse myself, and I was walking across the room from one side to the next. I don't know if it's easily visible, but we're going to see one thing that can determine the velocity of one of these action potentials is whether or not the axon portion of a neuron is heavily myelinated. Remember, myelin is coming from either the Schwann cells or those oligodendrocytes, and cells that are heavily myelinated like this are going to allow these electrical signals to move a lot more quickly versus these axons that are not myelinated at all. There was a video, moving video of this in the Google Slides that I sent out. This ended up being kind of a washed out picture, and I don't know if you can quite make it out, but these are those Schwann cells that are wrapping around the axon portion, and the ion movement, sodium coming in and potassium moving out, can only occur in those nodes of RANVA, in those little gaps in between those Schwann cells. So ion exchange only happening in those little gaps was like me taking these big steps across the room, and anywhere my foot touches the floor, that's where ion movement occurs. So taking these big steps, we can see ion movement doesn't occur everywhere across the surface of the floor or the surface of the plasma membrane, but only in these little gaps. Versus the unmyelinated axon, where I was doing this last time, where the electrical signal is going to have to touch every spot on the floor to get all the way to the other side. And this is going to take much longer for me to get all the way over there versus taking those big steps. So whether there is myelination present or not is one thing that plays a role in how fast this action potential can move along the axon. The final thing, which was on this slide, has to do with the diameter of that neuron. We're going to see that the larger the diameter of a neuron, the faster that action potential can travel along it. Versus the smaller the diameter, it's a slower signal. My analogy for that is, imagine, you see this sometimes, like they put them in drainage ditches or something. Like imagine just a big gigantic PVC pipe that has a diameter of like 6 foot. So if it's this huge PVC pipe, we could all just walk through the middle of it without touching the sides. And we can easily, just as a class, walk right through this PVC pipe to the other side, as long as that diameter is big enough. But if the diameter was only like a 2 foot diameter, we would each be maybe wiggling through there like this, and it would take a lot longer to wiggle through that narrow tube. We would be scraping the sides, and that friction as we hit the sides would slow us down. Same thing happens with these neurons. There's less friction in larger diameter neurons, so the flow of ions can happen more rapidly. more fast. Maybe I'll edit that out. I'll probably forget. All right. That was kind of a quick run through of the neurophysiology slides. And I say quick because half of the stuff we had set up last time with our diagramming of an action potential, separating an action potential versus a graded potential, and where those occurred on a neuron. So I'll leave that up. Maybe we can spend a couple of minutes going through any kind of questions that you have about this. Because on the other side of this, what we're going to do is we're going to kind of switch gears. It might be refreshing to pull our heads out of the physiology part of this and just once again kind of map some anatomical structures. We're going to look at brain and maybe spinal cord models if we can get that far. I just want to point out all of these things from the lab list so that you can pin them, you can practice finding all of these things so we can take our practice practical next time. And then you'll see it. Once we know all the structures of the brain, then we can on Thursday talk about what each of those parts of the brain do. I don't know if you... Oh, I haven't passed out new handouts for parts of the brain yet, so I was holding on to that so that we could maybe think about any of these neurophysiology questions. I'll get this out of the way for a second. Okay, sodium-potassium pump, we talked about how that works. Again, let me know if there's questions about any of these particular diagrams. I just wanted to point out a few things initially, which is what we're looking at on the second page. I just put a typical neuron. You might recognize this as a multipolar neuron. We've taken a cross-section through the dendrite and we've zoomed in on it. And what I was hoping that you would write in here, I've erased it, but it was the diagram of a neuron that we had originally drawn over here. So inside you can just put something along here. You could just use words and say high concentration of potassium, low concentration of sodium. And then there's a bunch of those negatively charged anions, which is what causes the cytoplasm of this neuron to be negative 70 millivolts. And we said outside the cell, everything in blue here is our extracellular fluid. There was a high concentration of sodium and a very low concentration of potassium. So this is where we were kind of setting up that imbalance where it's going to be very easy for sodium to rush into the cell. And also it's going to be very easy for potassium to rush out of the cell if those potassium gates open up. So again, inside the cell, high potassium and low sodium. Outside the cell, it's high sodium and low potassium. This graph is kind of graphing all those different parts of the action potential that we talked about. Hopefully that's clear. I do have the key over there if you want to check any of these things out. On this page is where I was trying to make the distinction between the fact that those sodium voltage gates open very quickly and they also close very quickly. But the voltage-gated potassium channels, they open slow and they close slow. Here are some, I think this page and the next two pages are also kind of confusing. So I wanted to at least stop here for a second. Where we are at now is we're at the axon portion of the neuron where the signal is always going from the axon hillock down the axon towards the axon terminal. So we're in this part of the neuron, and all I was trying to show with this series of pictures is that the blue part of this graph, even the D right there, tells you that this is depolarization. The colors don't match up with the colors I've used here, but you can see positive ions moving into the cell. We know positive ions moving into the cell. That's depolarization. The cytoplasm becomes more positively charged in that area. And the second picture here is just following this action potential as it ripples down the axon towards the axon terminal. So red here we have a resting membrane. The resting membrane first depolarizes. Here we are. We've got a resting membrane. It first depolarizes, and then following depolarization is always some type of repolarization where potassium ions leave the cell. And then following repolarization, we're right back at our resting membrane potential. So sodium positive charges move into the cell. This is depolarization. depolarization is always followed by a repolarization, a purging of those positive charges back out of the cell. So in this case, what's being purged are potassium ions out of the cell. The green is repolarization in this case. And then if I go down even further, this is just a little bit later in the diagram where you can see this action potential continues to move towards the axon terminal. You've got this wave basically of sodium ions that move in, followed by this wave of potassium ions that move out. And that wave of ion movement as it ripples down this axon is what we're describing as this electrical charge. We were seeing it in that video as that little beam of light that was shot by a neuron. That beam of light is this wave of sodium ions moving in and potassium ions moving out. So you can see in the color version of this picture, blue arrows represent the movement of sodium ions into the cell. Sodium ions cause depolarization. And then the green arrows represent potassium ions moving out. We know potassium ions moving out is responsible for that repolarization. So just a lot of practice with depolarization, meaning sodium ions moving into the cell becoming more positively charged. And a lot of practice with this repolarization dealing with potassium ions leaving the cell and the cell becoming more negatively charged. This is the same thing, but it doesn't have colors associated with it, so it's a little bit more confusing. These little gray areas represent Schwann cells, and we only can have ion movement in between those spaces in these nodes of Ranvier. And here at the initial time we've got, see with this picture we don't know where the axon terminal is or where the axon hillock is located. We don't know what direction initially these signals are being sent. We've got resting membrane potential, and then one segment of this axon depolarizes. We can see here in the next, as time progresses a little bit, the second segment of this node of Ranvier depolarizes, and the one that was just depolarized had repolarized. And if I go down a little further, we'll see that that wave of depolarization continues to move. Actually, that's the question here. This is, I guess, what you should be thinking. If we're looking at these little spaces, this one was just at rest, but what might you expect this one to change to next? If we can see how our wave of...
Depolarized?
That's right. So this one is going to be the next in line to depolarize. We can see that depolarization started on this end of the axon. It's making its way over here. So we're kind of figuring out that the cell body is over here, and the axon terminal is going to be over here because the action potential is now headed towards me. So this would depolarize, and the segment that had just depolarized would what follows our depolarization? That's right. So down here, this segment would now be repolarized as this one depolarizes, and the segment that was just repolarized, what follows repolarization, is once again our resting membrane potential. So just another way of kind of graphing that this electrical charge that we are saying these neurons send is just a wave of sodium ions moving in followed by potassium ions moving out. Depolarization followed by repolarization. And then what I think is maybe the best picture in this class handout, I would put a star next to it, and that's describing this interaction between a presynaptic and a postsynaptic neuron. You'll notice it's almost identical to the same thing that you had in the last exam where we were talking about a neuromuscular junction, except instead of these non-specific ion channels, these chemically gated channels being located on a motor end plate, that motor end plate was on one of these muscle fibers. We're not talking about the motor end plate. Instead, we're talking about one of these other postsynaptic neurons. So this time, those chemically gated channels are located either on a dendrite or on a cell body of another neuron, but they behave the same way. If a chemical is located, if it's docked to that protein, I get my pointer here, you can see those channels are going to open up. When they open up, we describe them as non-specific ion channels, so you can see sodium coming in and potassium going out. That ion movement continues as long as that chemical gate is open. But as soon as the chemical signal is removed, we can see this chemical gate closes. There's no more ion movement. And then the sodium-potassium pumps would reestablish that resting membrane potential. I guess we'll wait a few minutes for the other people to show back up, and then we'll get started. I'm going to start with that concept map of the brain, and I'll really try and just point out all of the parts, the anatomy stuff first, and then we're going to kind of get into the physiology. You can see maybe as deep as we go in terms of physiology of the brain is just to relate the different parts of the brain and what they do. So there are some matching questions. I guess it's really the second page of this handout, where we kind of match some of the brain functions with the location. So we'll first map out, where is the medulla, where is the midbrain, where's the pons, where is the thalamus, and all of that stuff. And then we'll talk about what each of those parts do. And I'm sure a lot of the questions that you'll see in the next exam are a lot of these scenario-based questions where you're in the emergency room and the patient that's coming to you is displaying some type of situation. And depending on what's abnormal, that's going to clue you in as to what part of the brain might be damaged. We do a lot of that with brain function, this unit, but where we will go in, just to preview Unit 5 for a second, in Unit 5 we do these cranial nerves, the 12 pairs of cranial nerves, and we'll talk about how they do head movement and eye movement. And it's one of the things that you check for, cranial nerve damage, when you're assessing somebody, making them follow the light left or right, up or down, or tilt their head a certain direction. You're checking to see if there's damage to any of those cranial nerves, that they're not able to shrug their shoulders or stick out their tongue or look down or any of those things. You're assessing for cranial nerve damage, just like we will be assessing for whether or not parts of the brain are functioning correctly. So I guess here we are. It looks like we're about to get started. So I've got one of these concept maps that you can see it's kind of broken out nice and symmetrical, where right in the center we're going to put the brain, and then off of that we're going to see the brain is going to be separated into 4 sections, and then we'll look at each one of those 4 sections a little bit more closely. So you are just in time. Y'all should have one of these, the concept map. So as you get settled, I'm going to start with my diagram here of the parts of the brain. And then once we map them out, we're going to go through and kind of assign a function to each one of those parts. I'm sure I'll have to use maybe both of these parts of the board. We'll see how well I can fit it in here. I'll just put brain right in the middle. And we'll see that the brain is going to be separated into 4 distinct parts. And if we just start at the 2 parts that I've listed at the top, they kind of correspond with one another. We'll call one part of the brain the cerebrum. So cerebrum it's going to make up about 80% of the brain. When we're thinking about the cerebrum, we're talking about this kind of tan-colored part of the brain. All of this stuff that I'm holding on to, again, it makes up about 80% of the brain. This is our cerebrum. Versus this little, maybe the 20% of the brain, this little different colored back portion of the brain that we're going to call the cerebellum. If I talk too quickly, I'll mix those up today, but keep me straight. Cerebrum, we can see, is this, like I said, the 80% of the brain that you see when you're looking directly on top of it. And this entire cerebrum we can separate into 3 different parts. There's going to be the most superficial part. And look at this model. If I take it apart, I'm going to hold up just a portion of this tissue. And you can kind of see a lighter staining patch in the middle. And then the very edge, maybe just the outer little 2 to 4 millimeters of that, you can see it's kind of a little bit of a pink edge. Again, me holding it up at this distance is not going to work for anybody in the back of the room. But when you grab this cerebrum and you open it up, you'll see the very outer 2 to 4 millimeters is going to be what we call the cerebral cortex. cerebral cortex. Then in the middle, there is what we call cerebral white matter. And then if we go deep within each cerebral hemisphere, we're going to once again see these collections of cell bodies and nuclei that we're going to call cerebral nuclei. I can't spell. Nuclei. This term cerebral nuclei, another term for cerebral nuclei is basal nuclei. I can't spell again. So basal nuclei and cerebral nuclei are the same thing. Nuclei, when we were talking about the brain and spinal cord, we said the term nucleus or nuclei are going to refer to a collection of cell bodies and dendrites in the brain and spinal cord. We also said that a collection of dendrites and cell bodies in the brain and spinal cord are not going to be covered in myelin. I'm looking for one of these cells that shows myelination. In cell bodies and dendrites, there is no myelination. So anywhere there are cell bodies and dendrites exclusively, these are going to be areas of gray matter. White matter just is an indication of multiple layers of myelin. And we also know that anywhere there's myelin, we're just talking about the axon portions of those neurons. We're not talking about the cell bodies or the dendrites. So I just wanted to come back over here and maybe put a note that this cerebral cortex, I'll just list it down here, this is gray matter. We know that's cell bodies and dendrites. Cerebral white matter, it's right there in the name. These are going to be heavily insulated axon portions. And then cerebral nuclei, anytime we see that word nuclei in the brain and spinal cord, it's describing a collection of cell bodies and dendrites. So looking at this superficially, the cerebral nuclei is going to stain in kind of a gray color. This is also gray matter.
You said cerebral white matter is heavily insulated.
Yes, and I'll start with some slides that show this. I just didn't want to cut off what we're drawing. I tried to show that if you look at some of these models, like the cross-section through this part of the brainstem falls off there. But I've cut through the cerebral cortex, and you can see this white matter that kind of makes up the middle portion of the cerebral, well, this entire cerebrum. The outer edge is what we're calling the cerebral cortex or the gray matter. It's actually kind of a pink color on this model, but you can see the white matter, they've kind of used a white color to indicate that. What else? If we want to go ahead and list functions with these things, the cerebral cortex, this is where you're going to store memory. So we'll say memory storage.
Both long-term and short-term.
Yeah. So cerebral cortex is where we store our memories, and I'm going to show some pictures of a second, if I haven't already shown those at some point this semester. Did we ever look at the internet to compare the cerebral cortex of different mammals? If we haven't yet, it's interesting. Cerebral cortex, if you look at the cerebral cortex of humans, let's just grab one of these brains, we can see the surface of our cerebral cortex. I'll just hold half of this brain up. We can see has a lot of these little raised ridges that we're going to call gyri, and these little crevices that we're going to call sulci. But all of these ridges that we can see, kind of this wavy, marbly looking structure, this increases the amount of surface area in the cerebral cortex. Let's do it this way. That's a good one. Pull this thing down for just a second. So here we're looking at just the different cerebral cortexes from several different mammals. You can see, get out of the way and grab my pointer. In the case of something like a rabbit or a rat right there, you can see they have a very smooth surface on their cerebral cortex. And it's only the first like 2 to 4 millimeters deep on the surface of that cerebral cortex do you have a bunch of these neuron cell bodies whose job it is to store a bunch of memories. Now, if we compare that to something like a cat, you can see how a cat, they have these gyri and these sulci that form these collection of ridges that really increases the amount of surface area on a cerebral cortex. You can see a sheep or a chimpanzee or even a human, they have an even larger number of these gyri and sulci, so that this is, what it's creating is even more surface area, even more room to store memories. And you can even, this is why it's believed that dolphins are as smart. I mean, we have different capabilities. Dolphins can't work an iPhone. But if you actually look at their cerebral cortex, they have a tremendous amount of memory storage. In my analogy, I always use weird analogies for everything. The cerebral cortex can be like the little flash drive that you use to store PowerPoints or whatever. This smooth cerebral cortex might be a little like a flash drive that's a 1 gig flash drive. You know, you can store a few files on that, but it's going to fill up relatively quickly. And then as we see these increased number of sulci and gyri increase the surface area on the cerebral cortex, this is more room to store memory. So this would be like a 1 gig flash drive or however big they are now. So cerebral cortex is a place where we store memories, and you can see comparing different mammals. Some mammals have more surface area in their cerebral cortex. Anyway, here's just another picture I'll show briefly of one of these transverse cuts through the brain. You can see this cerebral cortex, the outer 2 to 4 millimeters of these cerebral hemispheres, which are dark staining. Again, they're dark staining because there are just dendrites and cell bodies. There's no myelinated neurons. But once we get into this portion, this is all what we would consider the cerebral white matter. And this is made up of just bundles of axons that are heavily myelinated. And then as we get deep to the center of each cerebral hemisphere, you can start to see this gray matter appear again. These are going to be what we call those cerebral nuclei, or basal nuclei is an older term. They're collections of cell bodies and dendrites. So there is, once again, that grayish color. To maybe list functions there, we know memory is stored in the cerebral cortex. So these cerebral nuclei that sit deep in each cerebral hemisphere, what they do is they inhibit unwanted muscle movement. I'll say that again as I pull this up. These things, these basal nuclei or cerebral nuclei, their job is to inhibit unnecessary muscle movement. So, I'm going to grab this brain stem right by your foot. Have you ever seen one of my buddies, actually, he's younger than me, and he was a fantastic musician, and he had to stop playing music because he had nerve damage. And one of the things that started to happen is you can tell what part of the brain is going out based off of the symptoms. And one of the things, my buddy started to develop Parkinson's disease. So what's going on there is it's a part of the brain that starts to, it's where these cerebral nuclei are deteriorating, and they're no longer functioning like they should. Their normal function, like we said, was to inhibit unwanted muscle movement. But if that's failing, then you start to see this unwanted muscle movement. So somebody that has Parkinson's might not be able to control their muscle movement, and that's, again, indicating a part of the brain that is having to deal with damage, or neurons are dying. I've turn the screen off so that we can continue with our discussion of parts of the brain. We're going to see these same parts map out on the other side. So if we can go over here to the cerebellum. And again, when we're thinking of cerebellum, we're thinking of just this little posterior portion of the brain. And if we were to open up that cerebellum, we would once again see three parts. There is the cerebellar cortex. That cerebellar cortex is just the outer 2 to 4 millimeters of this cerebellum. If we look in the middle of the cerebellum, there is cerebellar white matter. This cerebellar white matter, on your lab list, it actually gets a special name. It's called arbor vitae. This arbor vitae, it just means tree of life, because if you look inside the cerebellum, you'll notice that that cerebellar white matter branches in a way that kind of looks like trees or branches of a tree. So I'm just grabbing this brain. We're looking at the opened section of this cerebellum, and we can see that cerebellar white matter that forms those branching tree-like structures. But if we go to the either half of the cerebellar hemisphere, deep in each cerebellar hemisphere is cerebellar nuclei. Cerebellar nuclei, just like the cerebellar cortex, are made out of gray matter. Cerebellar nuclei, we can list the functions of the cerebellum here as, we said it functions both in long-term memory, but also in helping you coordinate muscle movement. So balance, like coordinating muscle movement, being able to walk straight, having proper balance, and all of that, it involves the cerebellum. So long-term memory, balance, coordinating muscle movement. These are all jobs of the cerebellum. Haven't left any markers anywhere. Now let's drop down to the two parts of the brain beneath this. We've got cerebrum and cerebellum, which are most of the parts of the brain that you can actually see whenever I hold this up. To get to the other two parts, we need to look at this brain stem, and a part of the brain that's kind of hard to see when you look at the brain from the outside, because it's kind of in the center of each cerebral hemisphere. So there are these models. These are brainstem models, and we've basically just removed each cerebral hemisphere, and we've removed the cerebellum so that we can see these other two parts of the brain. You've got these little, what look like ovals on either side of this brainstem. That region is what we call the diencephalon. So diencephalon is going to be one region, those two big ovals that are on either side of this brainstem. And you'll notice each of these diencephalon regions is attached to this stalk that I'm holding now, which is our brainstem. Okay, each one has three regions, so I'll just tackle this diencephalon region first. The thing to remember about the diencephalon, the three regions, they're all based around what we call the thalamus. So the thalamus is going to be this oval-shaped, kind of a football-looking structure in the middle of the brain. It is going to have above this thalamus a region of the diencephalon that we call epithalamus. We'll see epithalamus is just above the thalamus. And then there's an area of the diencephalon just below the thalamus that we call hypothalamus. So diencephalon, like we said, it's all about the thalamus. We will see that the thalamus is basically going to be the router of information that comes into the brain. All of the sensory information that comes into the brain passes through the thalamus, and the thalamus has to decide, okay, what part of the brain does that information need to hit? So it's kind of the router of sensory information that comes into the brain. Some noteworthy things that we can add to the hypothalamus and the epithalamus. I'll just use this color. Maybe the easiest way to recognize the epithalamus in our lab activities and how I'll even describe it in lecture is that attached to the epithalamus there is going to be one of the glands that we mentioned called the pineal gland. So let me say this with words because I don't want to add confusion here. What I'm trying to indicate here is the pineal gland is attached to the epithalamus. So maybe I'll just draw the pineal gland and I'll draw it maybe attached right there to the epithalamus. I'm not trying to say that the epithalamus is the pineal gland. However, we can find the epithalamus by finding the pineal gland. It's always attached right there to the epithalamus. And we can also find the hypothalamus because attached to the hypothalamus is going to be the pituitary gland. So pituitary gland is going to be attached to the hypothalamus, just like the pineal gland is attached to the epithalamus. Be careful not to confuse pineal and pituitary gland. They're two separate glands. They're close together. They're both located in this diencephalon region of the brain. One is attached to the hypothalamus while the other one is attached to the epithalamus. If we're okay there, I'm going to move over here to the brainstem part to list the three parts of the brainstem. I'll start, I guess, here at the top with what we're going to call the midbrain. We've got the midbrain. Then the middle portion is called the pons. And then the last part of this brainstem is the medulla oblongata. So those, the midbrain, pons, and medulla, are all part of the brainstem. When we think about the brainstem, we're going to have to find this. We have several brainstem models, right? These things that I'm holding up are the brainstem model. And when we take these brainstem models apart, we should be able to point out the midbrain. There are some structures attached to the midbrain that we're going to know as the superior and inferior colliculus. We'll say superior and inferior colliculus. Superior and inferior colliculus, we're going to learn these are little bumps located on the midbrain that are going to house neurons that detect either sensory or auditory signals. Here I am kind of segueing into the parts of the brain and our lecture discussion next time where we list what each part does. Superior colliculus, I'll put this way, visual signals. Inferior colliculus is going to be auditory signals. Have you ever been walking down the street and all of a sudden hear like this bang or some kind of loud noise, and almost involuntarily your head turns that direction because you've heard a noise? That reflex, that auditory reflex, is coming in through this inferior colliculus. So if this is damaged, you won't be able to have that reflex. Visual signals are picked up by that superior colliculus. So sometimes like a flash of light or there's something flashy will catch your eye and you'll turn and look. And that visual reflex is this superior colliculus. The pons, since we're already into this, the pons, if we look into the pons, there are going to be nuclei located in the pons that are either called, and this is on your lecture handout, apneustic versus pneumotaxic. Again, these are in the slides. I'm just somehow getting into them early here. But the pons, it's going to have neurons that control respiratory rate. I'll just say control respiratory rate. So there are neurons in the pons that can either speed up or slow down respiratory rates. The neurons that speed up respiratory rates, we're going to call apneustic neurons versus pneumotaxic neurons that slow down respiratory rate. Then this medulla oblongata is a part of the brainstem that controls what we'll consider vital reflexes. Vital reflexes are going to include things like coughing and sneezing. I'm also going to say vasomotor. So this thing also kind of helps control heart rate as well. Our second run through this on Thursday is we're just going to be matching up the functions of those parts of the brain with the actual part of the brain. Here I want to just list the parts of the brain and then be able to find them on the models. So here we go. I'm going to grab this lab list to make sure I don't miss anything. We have the four major regions, the cerebrum. Here we are. Cerebrum with those two cerebral hemispheres. The diencephalon region that contains the thalamus and the hypothalamus, as well as the epithalamus. The brainstem with the midbrain pons and medulla. And then we've got our cerebellum with the three parts to the cerebellum. Here's the brain. Again, everything in kind of a light tan color is the cerebrum. I said about 80% of the brain mass. Then the cerebellum is this posterior portion. Then in green, everything in green here is our brainstem, and then the diencephalon region, kind of this oval-shaped interior part of the brain, is in purple. So we've got diencephalon, brainstem, cerebrum, and cerebellum. Here's our cross-section through the cerebellum that shows us that cerebellar cortex. I knew I would switch cerebellum and cerebrum up at least once. So this is, I said cerebellar cortex, but I'm pointing to cerebral cortex. So cerebral cortex is where we store memories, like we said. Here's all the cerebral white matter. And then we've got deep inside those cerebral nuclei or basal nuclei. These are the areas that are failing in people that have Parkinson's because they can't inhibit that unwanted muscle movement.
I have a question.
Yeah?
In the cerebral cortex, the textbook says the neurons there are interneurons. What kind of neurons are those?
Remember when we were thinking there are three functional classes of neurons: sensory neurons, integration neurons, or motor neurons. And integration neurons and motor neurons are all multipolar. So it would be a multipolar neuron, and it's functioning as an integration neuron because it receives signals, it processes those signals, and then decides the proper response out to a motor neuron. We said all interneurons, they're located in the brain and spinal cord. They're never outside of the brain and spinal cord. And some of those interneurons are not myelinated at all. So these in the gray area would be unmyelinated neurons, cell bodies, dendrites. If there's an axon, there are unmyelinated axons, and you could have unmyelinated axons in this portion. There's just nothing with myelination in the gray areas. Yeah, no problem. On the lab list, as well as in your lecture handout, there are different lobes that we have to be aware of that each cerebral hemisphere has. I mentioned the ridges, the little raised areas that I'm pointing to one of the raised areas here. This is a gyre, as opposed to one of the little crevices there, which is a sulcus. There's the central sulcus versus those ridges there, which are gyri. Here's a better picture. We can see this ridge that's a gyrus versus a shallow depression that we call a sulcus. There are these deep depressions that separate the two cerebral hemispheres. A fissure is just a much deeper separation than a sulcus. This is just a shallow depression. So if we go back to this picture here, we can again see those raised gyri and then those little crevices that we call sulci. But the other things we can notice from this colored picture of the brain, it has used a different color to separate the frontal lobe from the parietal lobe. And then we can see the occipital lobe and the parietal lobe. They're both kind of a blue color, but that occipital lobe is a little bit lighter color blue than the parietal lobe. Then we can see the temporal lobe kind of in a purple color or maybe a dark blue color. So 4 of these lobes of the brain correspond with the bones that protect that part of the brain. So the frontal lobe is protected by the frontal bone. The parietal lobe is protected by the parietal bones. The occipital lobe is protected by the occipital bones. These temporal lobes are protected by the temporal lobes. I said temporal lobe, protected by the temporal bone is what I meant to say. And then on these models, you can't really do it. But if I were to grab this temporal lobe and the frontal lobe and try and pull them apart, if I could separate them a little bit, what it would reveal is this 5th lobe of the brain called the insula. The insula, this 5th lobe, is between the temporal lobe and the parietal lobes. I feel like one of these brain models allows me to do that. And it looks like it's this one. So if I bring this one to the front of the classroom, you'll notice between this temporal lobe and our frontal lobe, I can actually kind of pry it open a little bit. And those gyri that you see, those raised ridges and then little crevices, the sulci, that's part of the insula, the 5th lobe. If I go ahead and take that out, that 5th lobe of the brain that we're referring to, that's really just the outer surface of this diencephalon. Remember, this was a brain stem model, and you can see those gyri and those sulci. So this surface of this brain stem model is also the insula, that 5th lobe of the brain. Okay, so we've seen the 5 lobes. We've made a distinction between a sulcus, which is a shallow depression, and a fissure, which is a much deeper depression. Let's now look at longitudinal fissure and transverse fissure from these sulcuses. Here's a better picture. So this is our longitudinal fissure. It separates the cerebral hemispheres into a left and a right cerebral hemisphere. So longitudinal fissure, let's see if we can see the... Nope, I'll go back this way. Looking for a side view. Maybe this one will have to do. There is a transverse fissure that separates the cerebrum from cerebellum. So between the 2 cerebral hemispheres, we have a longitudinal fissure, but between cerebrum and cerebellum, there's the transverse fissure. In terms of the sulcuses that we had to point out, a lateral sulcus and a central sulcus. That central sulcus separates the frontal and parietal lobes. You can see it runs right between, again, that precentral gyrus and postcentral gyrus. There's our central sulcus. And then the lateral sulcus is the one that runs between the temporal lobe and that frontal lobe. We had just opened up that lateral sulcus. As I peeled that open, you were able to see the insula, that 5th lobe. Okay, so we've pointed those things out. What else is there? I'd mentioned the function of that cerebral cortex is really just the surface, the 2 to 4 millimeters of gray matter on the surface of each cortex. It's there for memory storage. Like I changed the oil in my truck this weekend, and I had to remember where I left the wrench to take the oil filter off and all that stuff. It's all stored in the cerebral cortex. Those little outer portions. Let's think about the cerebral cortex for just a little bit before we get into any of the other parts of the brain. If we look back at a picture of that cerebral cortex, the colors in this slide represent the different functional classes of neurons. So this kind of ties into something that you were just thinking about. We were talking last time about neurons functioning in different ways. Some neurons function in processing signals like sensory input, signals towards the brain and spinal cord. Other neurons send signals away from the brain and spinal cord and coordinate movement, some type of skeletal muscle movement. And then all the pink areas here represent those integration neurons, or what are sometimes called association neurons. Association and integration, you can consider the same thing. These are neurons that do information processing. And you'll notice there's a couple of dotted lines that have been mapped out. These are areas of the brain that function in language. This first area is all within the pink tissue. So remember, these are neurons that process information versus the red neurons that are pulling the strings on some type of muscle, getting a muscle to move. Those are the motor neurons. This area is what we call Wernicke's area, and this area over here is called Broca's area. Let me get those labeled. So now we can see a bunch of things on this slide. But what is labeled is this little dashed area within the association neurons. This is called Wernicke's area. We can describe the function of Wernicke's area. This is sometimes described as where you would have your individual personality. I mean, there's a lot of areas in the brain that can qualify for that, but maybe how you respond to certain jokes, like maybe the things that I find funny might be weird. Other people might find different things funny, but how you respond to certain jokes is partly a function of this Wernicke's area, because again, what you're doing is you're processing information that has come into you. You know, you're hearing the jokes. Is that funny? Is that not funny? And then how you respond has to do with this Broca's area. So Broca's area is outlined in red. So that tells us that it's going to coordinate muscle movement. So, you know, whether you laugh or whether you tell somebody that's a stupid joke or however you respond, this is carried out by Broca's area. Maybe another analogy that's sad but true. Wernicke's area, this is, like I said, an information processing area, and then Broca's area is like once you've done your information processing and you want to respond, you would use Broca's area to coordinate muscle movements to speak. And when I used to make trips to the nursing home and talk to my great aunt and relatives that were there, you could see sometimes when they had something that they wanted to say, but they just can't coordinate the muscle movement to say it. And that, you know, you can think about what's going on in the brain there. So one part of the brain is functioning just fine. They are processing information. They're thinking of things. Some of them don't want to be in there, and they would plead for me to take them out of there, but I can't. But then sometimes, you know, not being able to form the words properly or say what you're trying to say is an indication that this Broca's area might be not functioning properly. So I just wanted to point out at least two of these functional regions of the brain. Again, everything in pink is information processing area. Anything in red is coordinating some type of muscle movement. Anything in blue is coordinating some type of sensory input. I won't distinguish that any further because that will be additional stuff. If we have time on Thursday, maybe we will. If there are questions about any of this stuff, throw them out there. I wanted to point out at least two functional areas of the brain which we've done. Wernicke's area, like we said, in Broca's area. There are other parts of the brain, but I wanted to at least give you an example of information processing versus a motor control function of the brain. What else is there? In addition to Wernicke's area, like we said, that has to do with recognition of speech, you know, like processing that information versus Broca's area that actually generates those motor signals and coordinates muscle movement and allows you to form words. So there's a lot of different functional regions of the brain. I feel like we mentioned at least two more in the second part of this PowerPoint. But I'll jump over functional regions of the brain to show just a couple of pictures. These are spatial representations of the cerebral cortex. So what we're looking at is a cross-section through the cerebral cortex, and we're really just focusing on the outer 2 to 4 millimeters, really that cerebral cortex. We're not worried about any of the cerebral white matter. But what this is showing is that there is a disproportionate amount of cerebral cortex that is set aside for coordinating movements, like in the tongue or in the facial expression or in the hands, versus look at how much of that cerebral cortex is set aside for coordinating muscle movement of the wrist or the elbow or the arms or the shoulders or the trunk of the body. I mean, there's only so many movements I can do with the trunk of my body versus look at how much look at how much cerebral cortex is set aside for coordinating muscle movements in the hands or facial expressions. I mean, our faces are just so expressive because we have so many of these slight little adjustments that we can make to different parts of our face that convey different emotions and ways of communicating. So we can see more of the cerebral cortex, the limited space of that cerebral cortex set aside for coordinating movements in the face and in the hands. Again, that's how we encounter the environment mostly. And then if we look at the sensory side of the brain, there's also a disproportionate amount of cerebral cortex that's set aside for picking up sensory information in the face and in the hands, as well as in the feet and genital regions as well. But again, not that much brain cerebral cortex set aside for monitoring sensory information in the trunk of the body or the elbow. We're not feeling around with the models with our shoulders or anything like that, but we are feeling with our feet and our hands and our face. So lots of space set aside for sensory neurons in that part of the cerebral cortex. Breaking away from cerebral cortex, finally, where all the memories are stored, I want to mention just briefly some structures in the cerebral white matter. We know that cerebral white matter, we're not talking about the entire neurons, we're just talking about the axon portions, and we know since it's white staining tissue, it's going to be heavily myelinated axons. And sometimes just the axon portion of a neuron is referred to as a fiber. So here's a word association. I know I'm using the same colors here at this point, but the axon of a neuron is going to be referred to as a nerve fiber. I just point that out because we're about to talk about these commisual fibers, association fibers, or projection fibers. And fibers, again, are just axon portions of neurons. Where they go in the brain are going to be different locations. So we can see all three fibers that we have to know in this one picture. Let me check to see how I'm doing on time so I don't get distracted here. The first fiber to point out are going to be these association fibers. You can see those in a pink color. Association fibers, you'll notice, they allow communication within one half of the cerebral hemisphere. So an idea that is in this cerebral hemisphere can use these association fibers to bounce around to other places within this cerebral hemisphere. So association fibers allow us to bounce around within a cerebral hemisphere. If these ideas are going to bounce to the other cerebral hemisphere, the only way for signals to get from one cerebral hemisphere to the other cerebral hemisphere is by going through this corpus callosum. This corpus callosum is going to be made out of a collection of commisual fibers. Commisual fibers allow these action potentials to go back and forth between both cerebral hemispheres. Now, so far we can see how action potentials bounce around in one cerebral hemisphere or even how they bounce around to the other cerebral hemisphere, but we have not yet got any of these electrical signals out of the brain and down to the spinal cord. The only way these electrical signals can get from the brain down to the spinal cord and out to muscles is through these projection fibers. So our third and final fiber to introduce are these purplish fibers. Those are projection fibers that go from the cerebral cortex down to the spinal cord. Commisual fibers, anytime you take a brain model apart and you can see this structure called the corpus callosum, which is where each cerebral hemisphere kind of anchors down to this corpus callosum. That corpus callosum, if we were looking at it face on, is just a collection of these commisual fibers that connect both cerebral hemispheres together. They allow the two cerebral hemispheres to talk to one another. Has anybody ever heard of those situations where, like, this is true, this is probably the craziest thing one of the craziest things I think that is real. My dad would tell me these stories. He worked at Scott & White for like 30 years. And he wasn't a doctor, but he wrote papers based off of, I guess the doctors would write papers and they would send them to him in editing to edit these papers before they get published. And he would see these crazy stories. And one of the things that kept happening, this was, again, in Temple, so these were a lot of soldiers that came from the Fort Hood area. But one, and this was multiple individuals that had a similar type of thing happen. But if there is damage to the brain that damages these commisual fibers, then you have two halves of the brain that can't communicate. Like thoughts are locked over here and can't get to the other half of the brain. And what this sometimes led to, it led to an individual that, it's not phantom limb. Phantom limb is like if my arm got cut off and I don't have my arm anymore, but I still feel pain, like I have my arm still. That's like a phantom limb syndrome. This is something totally different. This is the opposite of that. This is where you have these individuals that have a perfectly good arm, but for whatever reason, their brain starts to think, this arm, I don't feel like it's part of me. And this is real. These people that they start thinking, man, I don't know, it's creeping me out that this arm is touching me. I want it off. I want it off. And so there was a guy that was going to Scott and White. This was a decade ago. He was going to Scott and White saying, I got to amputate this arm. I just want it off of me. It's freaking me out. Again, it was damage in the brain, making one part of the body feel like it's foreign. And the doctors were saying, we're not going to amputate your perfectly good arm. So what this person did is they went and got a bunch of dry ice from Walmart or whatever, and they put it in the bathtub, and they submerged their arm in dry ice until all the tissue was dead. And then they came back to the emergency room, and the arm was so damaged that they had to amputate the arm. So a few months goes by, maybe half a year or so, and the individual starts thinking that the leg wants the leg off. And they're like, we're not going to cut the leg. Does the same thing with dry ice in a bathtub and damages the tissue. So there are all these papers in this research trying to figure out, what is wrong in the brain that makes people think that, part of their body is no, and again, it has to do, the brain is very interesting. How different halves of the brain will coordinate, for different parts of the body.
Did it not hurt him to burn his
I'm sure it did, right? And so something's going on there, that's.
But maybe like a pain signal or getting somewhere.
No, I'm sure it hurt, but it was a trade-off, I guess, between just
Really didn't want the arm.
dealing with that pain. Yeah, the psychological pain of having the arm was more than dealing with the pain, for a week or so while it healed. Very, very interesting situations. Again, none of that's on the exam, but it's interesting to how the brain, parts of the brain communicate. And if they're not able to send signals back and forth, it can lead to some of these situations like that. What's that?
What caused it?
It was some type of battlefield injury that caused, I don't know if it was IED or if it was some kind of, what was the original cause to the brain trauma, but it was a result of some type of brain trauma from battlefield. All right. Let's see. I'll try not to get stuck in side stories like this. But from the front view, today we're going to look at brain models. On Thursday, I'm going to bring a sheep brain in, and we're going to cut through it like a loaf of bread. And this would be us kind of flipping through the loaf of bread, and we're like halfway through the loaf of bread, and we're looking at the interior part of the brain. You can see the cerebral cortex. We can see the cerebral white matter. And as we get to the deep insides of each cerebral hemisphere, we run into these cerebral nuclei. We said cerebral nuclei and basal nuclei. That's the same thing. Basal nuclei is an older term. But these little pink patches, those represent it's technically gray matter, even though they've colored in a pink color here. Those would be our cerebellar nuclei. That's twice I've done it. I said cerebellar nuclei, even though I'm pointing to cerebral nuclei. Cerebral cortex, cerebral white matter, cerebral nuclei. Let's hope that's only twice. Now, there are certain little specific parts of the basal nuclei. I'll point them out on the model, but they did not make the lab list. I believe these just made your lecture handout. You need to know that these 3 structures are all part of this basal nuclei. This little inside segment is what we're going to call the globus pallidus. There is this kind of wall section called the putamen, and then there's this section called the caudate. If we could look at this from the side view, we would see how this stretches out a little bit. Let's see if I have that. A little bit. We're looking at a transverse view from the top, and you can still see those pink staining areas that are the cerebral nuclei. I said it right that time. So cerebral cortex, cerebral white matter, cerebral nuclei. Again, caudate, there's the putamen, and then the globus pallidus is further to the inside. What we're about to do, once we shift from talking about the cerebral hemispheres, we're going to end up talking about the cerebellum, which is back here. Then we'll talk about this diencephalon region. Everything in the purple color here, this is not part of the cerebral hemisphere, but this is that diencephalon region that's part of the brain, well, the middle part of the brain stem. Touches the brain stem. And right on cue, we're going to getting in towards that diencephalon region that sits right on top of the brain stem. We said it hovers around the thalamus, so I'll use my pointer. The thalamus is easy to recognize on any of these brain models. If you just grab one of these half brains and flip it over, there's typically this little, it's kind of a golden colored little dot. Can everybody see that little golden colored dot that's right in the middle of these models? That golden colored dot is not the thalamus, but it is a structure that's embedded right in the middle of that thalamus. This is a structure called the interthalamic adhesion. That little gold structure is the interthalamic adhesion. The thalamus itself is kind of this oval-shaped, almost like a football-shaped structure that sits right in the middle of the brain itself. You have this oval-shaped thalamus in both cerebral hemispheres. So if I go back to this picture, you can see there's one oval-shaped thalamus in this cerebral hemisphere, and then the other cerebral hemisphere kind of sits around the other thalamus. And then both of those thalami are connected by that interthalamic adhesion. That's this little golden dot that I'm pointing to that connects these 2 football-shaped structures. If I had 2 footballs held together with a little piece of super glue, the interthalamic adhesion, that would be the super glue, and the thalamus there would be the 2 footballs that I've super glued together. Okay. So there is the thalamus, this kind of oval-shaped structure. The thing that we need to recognize below the thalamus, there is this little funnel-shaped area that we're going to call the hypothalamus. I feel like I've pointed to that already. Maybe we haven't.
Wait, can you point to what?
So this purple, kind of a V-shaped or a funnel-shaped area, this is the hypothalamus. It's first off, it's below the thalamus. And the other clue that we're looking at the hypothalamus is we can see attached to the hypothalamus is the pituitary gland right there. So pituitary gland is attached to this hypothalamus. And then it's kind of hard to really make out, but the superficial rim, there's a little superficial edge to the thalamus. That superficial rim is what we call the epithalamus. It's kind of hard to see the epithalamus, but one clue that we know that we're close to the epithalamus is by finding this pineal gland. So in our chart somewhere over there, we said the epithalamus has attached to it the pineal gland that is easy to see. On any of these half brain, on this half brain, let's see, there is this little pink structure that kind of comes off the top edge of that thalamus. Can everybody see this little pink, that little pink streak that goes across the, so we're not worried about the purple one.
Right under the purple.
Under the purple one, that's right. So that pink one would be the pineal gland. I know this far up here is not good, but pineal gland is probably the best way to indicate the epithalamus. There's not much of the epithalamus present on these half brain models, but we can find that pineal gland. So here again, stripping away the cerebrum and cerebellum, we can see these oval-shaped thalamus. We can see that beneath the thalamus, that little funnel-shaped hypothalamus that leads to the pituitary gland. What else? Just kind of looking at this cross-section of the cerebral hemisphere, kind of face on, we can see those two footballs that I described as the thalami that is found in each cerebral hemisphere. They're both connected by that interthalamic adhesion. You know, at some point I'm going to come back to this slide just because there's something that we can see interesting right here that's really, I feel like we've mentioned it before, but we really start to see these ventricles of the brain. These ventricles of the brain we're going to refer to as these fluid-filled pockets of the brain, and they're there basically to help cushion the brain from the inside, but they also circulate that cerebral spinal fluid. We're going to see it runs through these ventricles, but it also surrounds the outside of the brain and the outside of the spinal cord and allows those to kind of float in a column of fluid. So that fluid runs through these ventricles and also runs around the perimeter of the brain. Anyway, we'll look at the ventricles in just a second. Still thinking about the diencephalon, kind of the thalamus. We said that oval-shaped area right here, we can list its function as routing the sensory information that comes into the brain. We said it has to process sensory inputs and decide where in the brain does that need to be directed to. The other thing you can see, the thalamus is going to help make up the walls and the floor of the third ventricle. If I go back to this picture, these are going to be what we call lateral ventricles. But as soon as we get into this pocket of fluid here that surrounds that interthalamic adhesion, this fluid that surrounds the interthalamic adhesion is what we call the third ventricle. Really, the walls of the third ventricle are produced, they're made by the thalamus itself. So walls of the third ventricle are made by this thalamus. We said it functions to relay that sensory information to the cerebral cortex. The hypothalamus sits below the thalamus, that little funnel-shaped area that the pituitary gland is attached to. We'll see the stalk that connects that pituitary gland to the hypothalamus is called the infundibulum. We'll end up drawing that, I think it's in the next semester. But going back a couple of pictures, that little stalk is the infundibulum that connects this pituitary gland to the hypothalamus. If we haven't listed the function of the hypothalamus yet, we can say that the hypothalamus is really kind of the boss of the autonomic nervous system. When we set up the nervous system, we said there was the sensory division that sent signals back towards the brain and spinal cord, and then there was the motor division. And that motor division was separated into the voluntary muscles, skeletal muscles, and then all of the involuntary muscles like cardiac muscle and smooth muscle. It's this autopilot, the autonomic nervous system, that controls cardiac muscle and smooth muscle. The hypothalamus controls that autopilot. It controls whether or not you're in fight or flight or in rest and digest. Other notable things about the hypothalamus, there are neurons there in the hypothalamus that can help regulate whether or not you are thirsty. Without using any numbers, let's see if I can just describe this. You've got neurons in the hypothalamus who have their dendritic ends just out into the bloodstream. And if I were to eat like a big salty bag of potato chips, then what would happen is that the ion concentration, I don't want to mess up your bottle of water. Maybe I'll have to bring this in as a demonstration next time. But, you know, like I'm not adding any more water to my body. And if I just started to add a big bag of salty potato chips, then it would the ion concentration of my bloodstream would start to be more concentrated with particles in.
Do you need another bottle of water?
No. It's just so much easier if I can visually show this. All that's really happening is that you're increasing the salt concentration without adding any more water to that. There's a certain point where the dendritic ends of those neurons can sense that the bloodstream is becoming too saturated with ions, whether that's a bunch of glucose because you're eating Halloween candy, or whether it's a bunch of sodium ions because you're eating a bag of potato chips. Whether it's glucose or whether it's these sodium ions, when they get to a certain concentration in the bloodstream, it's picked up by the hypothalamus, and it's one of the things that makes you feel like you're thirsty, so that you'll ingest some water and kind of dilute the concentration of ions that you've ingested. Anyway, maybe with a demonstration that could have been better. But the hypothalamus, like I said, it regulates whether you're thirsty or not. It also regulates whether you're going to sweat if you're too hot, or if you're going to shiver in order to cool off. So body regulation, whether you're hungry or whether you're thirsty, are all regulated by the hypothalamus. The epithalamus, the structure that houses the pineal gland, we can say that this pineal gland is going to produce what we call melatonin. Not melanin, that we said was the UV ray protector in the skin, but melatonin is going to be one of these chemical signals produced by that pineal gland, and it's going to help regulate your sleep and wake cycles. It's actually the molecule that's responsible for regulating your sleep and wake cycles so tightly that some days you may forget to set your alarm clock, but you still wake up at the same time, because your body is what you would say used to waking up at a certain time, and it's used to waking up at a certain time due to the pineal gland and the melatonin levels that are produced. Okay, so we've seen the cerebrum. We've looked at cerebral cortex, cerebral white matter, and the cerebral nuclei. We've seen this diencephalon, the hypothalamus and the epithalamus. So let's see if we can point to the brainstem as well as this cerebellum. Looks like the next place we go to is the brainstem. So everything in green is our brainstem. We should be able to point all of these structures out on any of these brainstem models that we're holding up. Things that made the list. Grab one of those lists. There is this rounded anterior part to the brainstem that we're going to call the pons. Let's see if we have that labeled. Here's our pons. Then beneath the pons is this portion of the brainstem that is still kind of, see it has a little expansion on the anterior side. That expansion kind of tapers and narrows once we get into the spinal cord. But right before the spinal cord, kind of the swollen part of the brainstem is what we call the medulla oblongata. Just above the pons. If you can think of the pons as kind of being right in the middle of the brainstem, you've got this area above the pons that we're calling the midbrain, and the area below the pons that we're calling the medulla oblongata. We mentioned the midbrain right there. If I'll point those bumps out on one of these brain models. You can see the midbrain between the cerebellum and the cerebrum. If you look right in between those two, you'll see these bumps. Those bumps represent the midbrain. I'll transition to the next slide in just a second. What we're looking at here, we're looking at the brainstem from the side. But if we could look at the brainstem from the anterior view, right from the front, like I'm looking at it now, it would look something like this. And I'll use this brainstem model to show you that you're looking straight on to a brainstem. So we can see these would be the thalamus from either cerebral hemisphere. We're not worried about the purple diencephalon regions now. We're thinking about the green brainstem regions. We can see this anterior portion that we're going to call the pons. Beneath the pons, this is that medulla oblongata region. Again, we can it's not green colored on this brainstem, but you can see an expanded portion of this brainstem just underneath the pons. So we can point out this medulla region. And then just above the pons, these are structures that are going to reach out and make their way up to the midbrain. If I can take this brainstem model and turn it to its side. So here we are now looking at it this way. from the side view, we can see that pons sticking out anteriorly. Beneath the pons is this medulla region. And then above the pons, now that we're looking at it from the side view, you can see those bumps that I referred to as the superior or the inferior colliculus. If we could actually keep this brainstem turning all the way to look at the very back side of the brainstem, here we are looking around at the very back side of the brainstem. And when you can look at the back side of the brainstem, you can see there are actually 4 bumps. There are 2 of those superior colliculi and 2 of the inferior colliculi. Collectively, all 4 of those bumps are called the corpora quadragemini. 4 bumps, corpora quadragemini. What we've added to that is that the superior colliculi are going to be the ones that pick up visual stimulus, and auditory stimulus are picked up by way of these inferior colliculi. I felt like here it is. Inferior colliculus picks up those auditory reflexes. Superior colliculus picks up those visual reflexes. And we'll list more of the functions on Thursday. I want to at least get to these parts first. So we've seen midbrain. Pons, we said, is also part of the brainstem that has those neurons that help control respiratory rate. So if we're thinking about the pons, we said there are 2 groups of nuclei there that can either increase respiratory rate. So this plus sign indicates that it's these apneustic nuclei that can increase respiratory rate. Or what we call pneumotaxic nuclei are also there in the pons, and they decrease respiratory rate. And then the medulla, we said this is the region that controls our vital reflexes, things like coughing and sneezing, heart rate, so cardiac rate, vasomotor is just blood vessel control. So a lot of these vital reflexes are controlled by the medulla, the neurons that live there. See, now the last section, which is the cerebellum, and then we'll try and find all of these things on the model. We're looking at just the cerebellum. You can now see that we're zoomed in enough to see the cerebellar cortex, all of that gray matter around the very perimeter of this cerebellum. We can see the cerebellar white matter, which kind of looks like this branching tree structure. So the cerebellar white matter is given the name arbor vitae. And let me flip this around. We're looking right through the center of this cerebellum. So now we've kind of just detached the cerebellum. We're looking at it from the outside. We can see both cerebellar hemispheres. And there is this structure that connects both cerebellar hemispheres. It kind of looks like the head of an earthworm. This structure right here is called the vermis. Here it is. It describes this structure that connects the 2 cerebellar hemispheres. To me, it looks like the head of an earthworm. We said the white matter in the cerebellum is arbor vitae. The cortex, that is the outer gray area. And if we can get to the center, this is an actual cerebellum. We can see that cerebellar cortex in gray. We can see that cerebellar white matter as these branching arbor vitae structures. And then right there, if we get to the deep center of the cerebellum, we can see those cerebellar nuclei. Maybe I could turn the light off and you could see a little bit more easily that that's darker staining tissue. That gray tissue tells us that it's no longer myelinated axons, but it's nuclei and dendrites, cell bodies. Let me see where we're going next. Those really are the 4 parts of the brain that we introduced. So the next series of slides takes us down into the spinal cord. And just as a brief introduction to the spinal cord, you can see as we go down the spinal cord from the neck, the cervical region, all the way down to the lumbar region, the amount of gray matter versus white matter in the spinal cord is going to change. So very little, a lot of gray matter when we get to the cervical region, and then it kind of narrows, it tapers off in the thoracic region, and then comes back in the lumbar region. let's see what else. I had described the white matter and the gray matter before as, again, any white matter are just the insulated axon portions of neurons, and then anywhere where we see gray matter, we know there's no insulation. It could be, just to do our word association stuff that we talked about last time, if we're thinking about nuclei, this describes collections of cell bodies or unmyelinated axons in the brain or spinal cord. I think I put these up on the board last time. Brain and spinal cord. If we're thinking about the central nervous system, the brain and the spinal cord, the term nuclei are going to refer to, like we said, just a collection of cell bodies and dendrites. That's the nuclei. If we think about just a collection of axon portions, a collection of just the axon portions of the neurons are going to be called a tract if this is in the central nervous system. So brain and spinal cord, these white areas we're going to be referring to as tracts. And the gray areas, like we said, are going to be nuclei. Jumping out to something that we'll see in Unit 5. In Unit 5, we talk about the peripheral nervous system, things outside of the brain and spinal cord, but we said the term ganglia and the term nerves also refer to parts of a neuron. So since we're diagramming these neurons, the term ganglia, like we said, is going to refer to a collection of cell bodies and dendrites out in the peripheral nervous system, and a bundle of axons out in the peripheral nervous system. Instead of calling that a tract, we're going to call that a nerve. Okay. Quickly, just ventricles of the brain. These made the lab list. I think we've pointed out all the parts on the diencephalon. I'm just going to catch up to the brainstem. I pointed out superior and inferior colliculus, the pons and the medulla. I'm going to go back to this picture right here. These darker green structures that are making their way from the pons up towards the midbrain, we can see them right here from the side view. These little stalks that are part of this midbrain, these are called the cerebellar peduncles. So on your lab list, there should be both of those. And I just see cerebellar peduncles. So this one is actually going up to the cerebrum. This is our cerebral peduncle. And these bands of fibers that we can see wrapping around, they would actually attach to the cerebellum back here. These are the cerebellar peduncles. So we have cerebellar peduncles and then our medulla oblongata beneath that. Getting to the actual cerebellum itself, this thing from the lab list, we can see our right and left cerebral hemispheres. We can see that vermis that connects the two. Cerebellar cortex is all of this stuff around the very outside, that dark staining tissue. And then cerebellar white matter, or arbor vitae, is all the light staining stuff in the middle. Now we're at the ventricles of the brain, or number 5. So I'm going to jump to these pictures of the ventricles. Here we are looking directly at the brain. We can see that each cerebellar hemisphere has its own lateral ventricle. These lateral ventricles are rather large, and they're kind of C-shaped. You can see how they stretch out all along each cerebral hemisphere. So each cerebral hemisphere has its own lateral ventricle. Notice how both of those lateral ventricles are going to drain into this structure called the third ventricle. In fact, I want to draw something real quick. Is it okay if I get rid of this thing? Yeah. Did everybody get a picture of that? This is just from the class handout, and I might be able to squeeze it in here. It's just a flow chart, or a little map that maps out how fluid goes through these ventricles. You've got most of these things from the lab list. If we're thinking about these ventricles, we can see each cerebral hemisphere has its own lateral ventricle. So I'll write, we'll say right lateral ventricle. So over here I'd have to put left lateral ventricle. I'll put a box around each of those lateral ventricles. We're going to see that fluid from each of those lateral ventricles, and I feel like we had this conversation the other day when we were talking about the fluid that is filling each ventricle. So I'm going to put inside each ventricle a little tangled capillary bed that I'm going to call a choroid plexus. Choroid plexus. This choroid plexus actually produces cerebral spinal fluid. So cerebral spinal fluid actually leaks out from these choroid plexuses. You would find a choroid plexus in each ventricle. So ventricles have these choroid plexuses that actually produce that cerebral spinal fluid. There are these two lateral ventricles, and then we said there is this third ventricle. This third ventricle, we said the thalami make up the walls to this third ventricle. So the third ventricle is going to have its own choroid plexus. But we're going to see fluid from both of these lateral ventricles. So fluid from the left lateral ventricle can drain into the third ventricle. Fluid from this right lateral ventricle can drain into the third ventricle. These little passageways that connect the cerebral spinal fluid from a lateral ventricle to the third ventricle are called the interventricular foramen. So interventricular foramen. This would be our left interventricular foramen, and we would have our right interventricular foramen. So it's through these interventricular foramen that cerebral spinal fluid goes from the lateral ventricles into this third ventricle. And then from the third ventricle, we can see there is a structure called the cerebral aqueduct that connects the third ventricle to the fourth ventricle. So there is a fourth ventricle. We can see that fourth ventricle sits down here between the cerebellum and the brainstem. And this little stalk right here that connects fluid from the third ventricle to this little pocket right here that's the fourth ventricle, that's our cerebral aqueduct. So I'll label this part cerebral aqueduct. The cerebral aqueduct connects the third and the fourth ventricle. Again, the fourth ventricle also has its own choroid plexus that's producing cerebral spinal fluid. But from the fourth ventricle, what we're going to see is this fluid is going to make its way down, a couple of places. Let's say from the fourth ventricle, one possibility is, I'm going to fork this thing off. We'll say one possibility over here, the other possibility this way. Fluid, as it leaves the fourth ventricle, it could stay in the central canal. Central canal of spinal cord. The other pathway, if instead of staying in the middle of the spinal cord, this fluid from the fourth ventricle can leave out of what we're going to call the lateral or median apertures. So lateral and median apertures are going to allow that cerebral spinal fluid to move into these subarachnoid spaces. Let's see pictures of that now that I've drawn it. There it is. And my lab list. So I was going through the ventricles. We wanted to see both left and right lateral ventricles, how they lead into this third ventricle. There is that little cerebral aqueduct that connects the third and then the fourth ventricle down there. And each of those ventricles, we should be able to find a choroid plexus. I'm going to come over here to this model. This model on a stalk is just a model of the ventricles, just a model of those fluid-filled pockets of the brain. And you can see, if I lift this thing out, you can see as I twist it around these C-shaped lateral ventricles. And then you can see in the middle what kind of looks like I don't know, maybe like a bird's face or something with a little beak right there. That middle pocket is our third ventricle, and then the last little pocket down here at the bottom is our fourth ventricle. Look at how if you flip this thing around, you can see those pink structures. Those pink structures, part of each one of these ventricles, that's a choroid plexus. So each ventricle has its own little pink choroid plexus that's producing that cerebral spinal fluid. Here's the same picture. We can see these large lateral ventricles. Each one has its own little choroid plexus that's producing that cerebral spinal fluid. So the little arrows just indicate the direction that fluid flows. It goes from these lateral ventricles down to the third ventricle, then down through that cerebral aqueduct to the fourth ventricle. And then from the fourth ventricle, we need to jump to this next picture, this one, that shows from the fourth ventricle, either that fluid could stay right here in that central canal of the spinal cord, or that fluid can actually leak out into these subarachnoid spaces. And it's here in the subarachnoid space that fluid really surrounds the brain and spinal cord. This is what allows that brain and spinal cord to feel like it's in the deep end of a swimming pool and just kind of floating in a column of fluid. Notice how there are these little arachnoid villi. These are places that allow cerebral spinal fluid to drain back into the veins. If I can just jump to this picture real quick. The red areas represent a choroid plexus. I'll use this picture, I guess, because a choroid plexus, this just shows where that cerebral spinal fluid begins. It's basically originating right here on the choroid plexus. It's leaking out of the choroid plexus, and it's starting to fill up these ventricles. And then that fluid, as it circulates through each of these little hollow pockets, it's got to have some way of, think of it this way. If we're constantly producing new cerebral spinal fluid, then this old cerebral spinal fluid has to have a way of getting back into the veins, getting out of these spaces. Otherwise, these spaces are going to swell and fill up with cerebral spinal fluid. We have a picture of an infant in, I think, one of the next PowerPoint slides that shows an individual that has hydrocephalus, which is too much cerebral spinal fluid on the brain because those arachnoid villi are not draining properly. And if you have too much cerebral spinal fluid in the brain, that puts pressure on neurons, and neurons don't like that. So too much cerebral spinal fluid is problematic, and also not having enough cerebral spinal fluid is also problematic, because if you don't have enough cerebral spinal fluid, then it's like we're not in the deep end anymore, and gravity is kind of causing some of these neurons to have to stand up on their own, and they don't like that pressure. Okay, so clearly I've gotten through these slides. The last things, well, it looks like we don't have too many spinal cord things on here. I guess I'll pull spinal cord model stuff into the next practical. But there are some things on here like the meninges that we have to know. Meninges are just protective layers around the brain and spinal cord. And I'll just say that the spinal cord model is the easiest place for me to point to these protective layers of connective tissue that protect the brain and spinal cord. The outermost layer is called the dura mater. We can see it as a tough, fibrous layer. this outer dura mater, if we could somehow peel away this outer dura mater, it would reveal underneath it this second layer of connective tissue called the arachnoid mater. And if you look at that arachnoid mater, it almost looks like it has spider webs that run through it. Can you make out those little arachnoid fibers? That tells us this is the arachnoid layer. And then if you look at the connective tissue layer that directly touches the spinal cord in this case, this is going to be the pia mater. So pia mater, arachnoid mater, and dura mater. If I go back to this slide, we said that the fluid here, this cerebral spinal fluid, fills up the subarachnoid spaces. So it's between the pia mater and the arachnoid mater. Going back to this slide, that cerebral spinal fluid would be in this space right here, between the pia mater and between that arachnoid mater, in this subarachnoid space. All right.